there is nothing which is stopping you from excelling your paper 2. I think, yeah. So we need to discuss and then we see which is stopping you from excelling your paper 2. So, I can't see what awesome pet is inside this egg. Oh my god, what's that? Click here to answer and hatch your own pet on Adapted Mind. What's that? Come answer more math problems, then hatch and customize. All right, it was just a small technical issue, né? But I think it's fine. All right, I think fine. Uh-huh. Paper 2. Guys, don't forget that. Thank you so much for joining us, guys. Let's just try to see that we smash paper 2. If you have any challenge, let me know. People who are on Zoom, let me know. You will tell me. You will tell me. So again, I have ID to join Zoom. There is no need for you to have ID. Yes. Just let me know if you have any question. On people who are watching from yes, I'm seeing someone from Orange Farm. All right, yes. People who are watching from YouTube, people who are watching from Facebook, people who are watching from TikTok. Please let me know if you have any question so that I'm able to smash it down. Paper 2 is easy. Why am I saying it's easy? It is easy because not too much of cram work. It's basically you have to know just a simple concept and then apply those concepts. So we have different questions we have organized for you so that they are going to guide us, and then we focus on our paper so that you are able to pass it. I've been seeing people saying that human evolution is a problem. Human evolution is a problem. Evolution is a problem. I think I'll talk about it. I'm gonna give it more time now. So I'm just gonna go through DNA, meiosis, genetics first. Then when I reach there, I'll give it extra time on that. All right, let's start without wasting time. It is M.Saidi as usual, so that you're able to smash the papers. Let's first start with the simple. Let's just do a reminder. Let's remind ourselves, simple, as we tried. Okay, division of a cytoplasm. Cyto means cytoplasm. We say it is cytokinesis. Sugar found or present in the nucleotide of DNA. We say it is ribose sugar. Position of a gene on a chromosome. We say it is a locus. Process during which DNA makes an exact copy of itself. It's called DNA replication. They're saying undifferentiated cell that may be stimulated to develop into any other body cell type. We said it is stem cells. Mendel's principle that states that organisms possess two factors that separate so that each gamete contains only one of these factors. Mendel's law of segregation. The evolutionary theory proposed that long period of species with no change alternating with a short period of rapid change. This one they like it too much, and then you find out that students always tend to not to focus on it, or not getting it right. We call it punctuated equilibrium. This is what you're talking about. Let me just look at it here. Where you is just here. This is what they are telling you. Punctuated equilibrium. Yeah, it's here. It's this one. So this is the one which has, when you look at it, they say that this one is gradualism. This is gradualism, while this one is punctuated equilibrium. What does it mean? It has periods of, if you look at it, this rate and this rate is the same. You understand? So from here to here, it changes rapidly, and then it remains the same. So if you look at this and this rate, they are different. They are totally different. But if you look at it, it goes slowly, changes slowly until it reaches like this. So this one is called gradualism because it takes a lot of time for it to what? To change from this point to that point. While this one, from here to here, it doesn't need too much time. It's just a rapid change. Then it maintains that period without changing. So in actually, or in other words, you'll see that the gradualism, it will require intermediates. From this point to this point, we have two intermediates. It keeps on changing slowly. I always give an example of people who use maybe they want to make their skin much more lighter. You don't just wake up in the morning and then see your skin is lighter. No, you just need to, you apply every day. After 1 week, then the skin, you will see that there is a change. But if someone goes before you do and then comes after, maybe after a week, they will see a very big difference between when they left you and when they found you. So but when who is doing it, you might not see it that this thing is happening. Yes, but people who will look at you, they will see that the change is what is there. All right. So writing paper 2 tomorrow. Is paper 2 tomorrow? Paper 2 is on what? On Tuesday. All right. They're saying that tangled network of DNA, that DNA and the protein located within the nucleus, we call it chromatin network. The DNA, the natural shape of DNA, we said it is called the double helix. The shape or in the cell cycle during which cell growth occurs, we call it interphase. Then we have another, we always start with these terminologies. Ne? Yeah, they're saying that the group of similar organisms that occupy in a particular place at a particular time, and they're able to interbreed. They have ability to interbreed, so we call it a species. So remember that you say B only. Yes, manipulation of biological process to satisfy human needs is by technology, so it's B only. Then they are saying that discovery of the structure of DNA, we call it, who discovered? Francis Crick, James Watson. Remember, the people who are involved in the discovery of DNA, they're saying who discovered the structure of DNA. These two were working together, so it is both A and B. All right, so that is simple. Simple. So let's start with the questions of DNA. Let me just give you this question. Let's do this question. Let's start with this question. Remember, we said that this is a process which occurs in the nucleus, specifically protein synthesis. So remember, protein synthesis is divided into two. It's divided into transcription and translation. Don't go to the paper without knowing to describe those two. I'm not going to repeat them. Just know that don't go to the paper without knowing how to describe them. Why? Because we either gonna bring transcription, translation, or protein synthesis. Sorry, transcription, translation, or DNA replication. So now remember, this is what? This is, let me just do a recall. This is DNA structure. You see it is DNA structure. These are amino acids. We always see them like they are balls or they are boxes. Yes, they can put it here like this. It's a box. So either it's a ball or it's a box, or it can be like this shape. You always see it that it is on a T. So it is, we call it what? tRNA. It's a tRNA, and then these boxes we call them amino acid. So this Y is amino acid. Where the translation occurs, we say this called a ribosome, and then this structure which contains the information from DNA, we call it a messenger RNA. And then now there is a confusion where students make mistakes. This is not a protein. This is still a polypeptide chain. If it is 50 amino acids and below, we call it a polypeptide chain. The bond in between is called it a peptide bond. Peptide bond. Ne? But the chain is called polypeptide chain. Yes. I hope you are getting it. So please don't confuse. There is no polypeptide bond. We don't have it. If there are many bonds, we call it peptide bonds. So in between here, so these are amino acids. So this chain is not a protein. We say this is a polypeptide chain. Please don't confuse that. Then we have this is a messenger RNA. This is transcription. It is in the nucleus. They can even ask you another question, that where is DNA located apart from the nucleus? So we can find DNA in the mitochondrion or in the chloroplast. So we need to know that we can find DNA in the mitochondrion, or we can find it in the chloroplast. All right. Just a moment. All right. We continue to the next point. So describe the process of transcription. Here you need to describe the DNA double helix. Look at it. Look at it here. Look at it here. DNA double helix. DNA is a mark. It's double helix is a mark. Unwinds a mark. Unzip or weak hydrogen bonds the mark. So we have 3 marks already here. DNA double helix unwind, weak hydrogen bonds break. Then to form 2 separate strands. To form 2 separate strands, one is acting as a template. So one strand is going to act as a what? A template. Use to form what? Messenger RNA. What does it use? It uses messenger RNA nucleotide. So that now a complementary strand is formed, whereby A goes with U and then C goes with the G. This messenger RNA, I said that we can ask you this. What is the function of messenger RNA? It has coded information. Yes, it has coded information. What is my pen doing? It has coded information for protein synthesis. So if it has coded information for protein synthesis, then it means that that is the function of the messenger RNA. So it passes through this, what you call the nuclear pore. So if they ask you, what is this membrane? This is the nuclear membrane, and then this hole is the nuclear pore. Yes. So basically, you need to know that. So it aligns itself on the ribosome. So if they ask you a question that, give the organelle, there are 2 organelles in the nucleus, sorry, in the cell, which we usually ask you here. That is the mitochondrion, sorry, that is the nucleus and the ribosome. Those are 2 organelles. We have other organelles, because an organelle here we mean a double membrane structure, but those are the 2 organelles we usually ask you. So when they ask you which organelle, think about a nucleus, and you think about the ribosome. So ribosome, you will see that it has the what? It has the messenger RNA, which is located there. So if this is called X and ask you to describe process X, make sure that you identify it, that that is transcription. That will be the first mark for you to get. All right. So after that, we have another process which we call the translation. Translation means formation of a polypeptide chain. This is what I mean, formation of a polypeptide chain. While transcription means formation of a messenger. Yes, formation of a messenger. Describe the process which result in the formation of messenger RNA. So it means that now they have not told you the process. So it means that you have to tell us which process is that. All right. So now, if we try to explain this, you tell us that according to the messenger RNA codon, so therefore you need to know what is the codon. You need to know what is the codon. The transfer RNA, this transfer RNA with the complementary anticodons, the 3 bases on the messenger, we call them anticodon. The moment they match, this matches with this, then now the amino acid will be brought. The transfer RNA will bring the required amino acid to the ribosome. This is the ribosome. Yes, now the amino acid, this amino acid and this amino acid are joined together by peptide bond to form what? To form a required protein. So this is the polypeptide chain which is growing, which is going to form a required protein. So basically that's what it is needed. So you need to know that these are called peptide bonds, required protein. And where does it go? It goes to the ribosome. The codon must match with the anticodon. That's what we want. And then a specific amino acid should be brought, or a required amino acid should be brought. So this process, you just have to memorize it. So if we go back to our question, what is X? You see, I said it can be a box, it can be a what? A circle. So that is amino acid. Amino acid. And then what is Y? Y is anticodon. So when I'm answering this question, even in your mind, you must be very fast to answer that question. If you see that your answer is different from mine, then you start to ask yourself that, oh, I still need to focus on this more. All right, then we go. What is Z? Z is a structure, you see? Yes, remember it has the U. So it is a messenger RNA. All right. So give the nitrogenous base of DNA. Now, remember, this is Z, which is messenger RNA. You understand? So this is DNA. So to convert this to this, I'll just need to convert it once. So they're saying that the triplet middle. So I need to find out which one is middle. That is 1. That is 2. That is 3. So they want this one. Meaning that I'm going to say AUG. So I need to convert this into DNA. A goes with T on the DNA. U goes with A and G goes with C. So it means that it's going to be TAC. All right. Then they are saying that identify nitrogenous base. They want it on the Y. Where is Y? Let me check. Where's Y? Where's Y? Where's Y? Oh, they want it here. They want the nitrogenous base. Still, the middle one, they want, they're saying that give the nitrogenous base sequence of Y. So now we have to come back here, meaning that Y is the first anticodon, meaning that this is going to go CAU, C goes with the G, A goes with the U, yes, and then U goes with A. So it means that this is, meaning that I'm converting this into anticodon. Basically, remember, we have code, which is DNA, we have codon, which is messenger RNA, we have anticodon, which is transfer RNA. You have to know that. If you don't know, because sometimes when you are bringing these questions, we bring them in a different scenario. All right, they're saying that Thank you so much, Cheli, Tele, Boka, Bonga, Bonging, I don't understand the name. All right. We are saying that, um, name and describe the process that is shown in the in the formation of the protein. You see now this question, they are asking you which process is this. The moment you see transfer RNA and you see messenger RNA, definitely that is translation. Translation. You saying translation is a mark. So now you are going to describe translation. According to the messenger RNA codon, the transfer RNA with the complementary anticodon, bring the required amino acid, the ribosome, the amino acid are joined together by peptide bond to form a required protein. You can redesign it the way you want, but as long as these things in different colors are there, because that's what we mark. You just have to use your words to connect them together, and the process is a process. A process is a process. You know what it means when we say that it's a process. So you don't mess up the process. You don't first go to the toilet and then you eat. You first eat, you go to the stomach. After that, you go to the toilet and you pee. All right. Why? Because it is a process. All right. Let's go to the next question. So that one, you have to describe it. Mutation. They're saying that the table below shows that. Now you see, if you don't know what's the meaning of anticodon, you are gone. That's why I say that you need to know them. You need to know them. Code for DNA, codon for messenger RNA, anticodon for transfer RNA. So they're saying that the table below shows the codon for the same amino acid. Look at it. For some amino acid, not same. A mutation caused DNA base triplet to change from CTG to okay, I'm saying that I have CTG went to CTA. A mutation, remember this is on DNA. So and what is here is codon, which means a messenger RNA, meaning that I cannot read it direct. I need to know what does this indicate, what does it mean, and what does this mean. So C goes with G, T goes with A, and then G goes with C. And then this one, I need to translate it also. You have to write it there. Say C goes with the G, T goes with A, and then A goes with U, because it is a messenger RNA. Messenger RNA. This is DNA. This is code, and then this is codon. So if it is a codon, so it meaning that now it is corresponding to this. So I'm gonna read this. C GSC 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 GAC. It's here. Aspartic acid. Meaning that is aspartic acid. If it is aspartic acid, and then what does this one? GAU. GAU. Again, it is aspartic acid. Meaning that what does it mean? It means that just a moment, guys. Aspartic acid. Someone is saying that he's still confused with the bases. Bases. You have to know that A goes with the C, C, G. Sorry, what am I saying? You have to know that A goes with the T and then G goes with the C. This one is I call it a Bible, a Quran for, yeah, to guide you. But this is DNA. But if it is DNA to RNA, remember RNA, they don't have T, they have U. So now you just put a U there. So now you know whenever you see A, you go with the T if it is DNA. Whenever there is A, it goes with the U if it is RNA. So now if it is RNA to RNA, then it means that there will be UU. So it means that make sure that in your statement there is no T if it is RNA. Make sure that there is no U if it is DNA. Yes. Ah, all right. So we said that this is aspartic acid and also this is aspartic acid. So what does it mean? It means that the protein will not change. So you're going to tell us that this codes for this corresponding to aspartic acid. So correct. Then this codes for this, which also correspond to aspartic acid. So therefore, the amino acid are the same. The protein will not be changed. Yes. So that's how you can try to make sure that your description, your words, it must talk about that this is translated to this. You need to translate that this translates to this. We need to see this, the translation. And then we also need to see that this is translated to the messenger codon will be like this. And then now you need to tell us that this, what amino acid does it code for? And this, what amino acid does it code for? If the amino acid X, this amino acid is aspartic acid, and this one is aspartic acid, as we have seen, it means that the protein will not change. But if the amino acid, is different, for example, the first one is aspartic acid, the second one is valine, then it means that it will result in the change of a protein. Yes. So please take note of that, guys, before you go for the paper. We got the next question. They're saying that tabulate two differences between DNA and RNA. The question is not like that. They're saying tabulate two differences between DNA and RNA. RNA nucleotide. Don't make a mistake and put DNA is double-stranded. They are not asking you DNA and RNA strand. They are asking you DNA and RNA nucleotides. They are not saying that give the difference between this structure and RNA. No. They are saying give a difference between the nucleotide of DNA and the nucleotide of RNA. Therefore, you're going to tell us that DNA has deoxyribose sugar, RNA has ribose sugar, DNA has thymine, while RNA has uracil. And they say that make sure that you tabulate. Guess what? Just drawing that and you say DNA and you say RNA, even if you don't put anything in the table, we give you a mark. So guys, don't miss these marks. And guess what? Other people are going to lose this mark because for them, they have just drawn like this. They just draw like that and then they start writing here whatever they want to write. They start writing here whatever they are writing. It's wrong. You're going to cross it. So make sure that it's a proper what? A proper table. Ne? Yes. Differences between DNA and RNA. DNA, RNA. Cool. Then you tell us the point here. DNA has deoxyribose sugar. RNA has ribose sugar. DNA has thymine. RNA has uracil. Correct. Then we're gonna give a mark here. We 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 give a mark here. So make sure that you don't mix mangoes with oranges. If you say DNA has thymine, don't tell us that RNA has deoxyribose sugar or ribose sugar because thymine is a base and then the other one is a sugar. So don't mix. Mangoes must go with mangoes. Oranges must go with oranges. Like that. All right. So we continue to the next question. We continue to the next question. Tendani, if you can make it a little bit what? Reduce the speed. Yes. All right. We continue to the next question now. We got the next question. So that is a simple question. Another question maybe describe for me the process of as describe for me the process of, no, the structure, describe the structure of DNA. 6 marks. DNA is double helix. You'll just look at the DNA and then you start telling us what you see on the DNA structure. Basically like that. What do you see really with the DNA structure? Double helix, it has is made up of nucleotides. Each nucleotide is made up of a deoxyribose sugar. It has a phosphate group. It is made up of 2 strands. What do you see? These 2 strands are joined together by weak hydrogen bonds. Guys, these are weak hydrogen bonds. Don't call them anything else. Weak hydrogen bonds. That's it. Yes. Then from there, describe, tell me what is DNA replication. DNA replication is the process by which DNA makes the exact copy of itself. Where does it occur? It occurs in a nucleus. When does it occur? It occurs during interphase. Why does it occur? To double the chromosome number. Yes. How does it occur? You describe the process of DNA replication. Yes. So please make sure that you go when you know these what? When you know those questions. What is DNA replication? When does it occur? Where does it occur? Why does it occur? And then how does it occur? Which means that you describe the process which is being shown up there. All right, let's continue. Before we continue, let's go for a tea break. Tea break is terminologies. The hydrogenous base found only in RNA. Now we know, I think. It's easy. It's easy. Let me know in the comment section. The use of biological process, organisms, or system to improve the quality of life. Let me know from your comment. The type of variation in population in which someone was asking me a question. The type of variation in which a population within a population which there is a range of intermediate phenotypes. Yes, okay. Someone said uracil, correct. Meaning that you got that answer correct. So this is called what? It's called biotechnology. All right, correct. So here we are saying that if you call it complete, intermediate, it's incomplete dominancy. All right, upright posture and walking only on 2 limbs, bipedalism. Sugar found in a nucleotide of DNA. Sugar found in the nucleotide of DNA. I don't know it. Please let me know from the comment section. Genetic disorder resulting in abnormal clotting of blood factor or blood clotting factor, meaning that you are not able to produce that blood clotting factor. You don't have ability for the blood to clot. We call it hemophilia. Hemophilia. Yes. Study of fossil, paleontology. Paleontology. Yes. Someone is saying, give me answer here. What I saying? Can you repeat it? Repeat what? So which um they're saying that present day distribution of living organism across the continent. How do you call it? Present day distribution. Someone saying hemophilia, deoxyribose sugar. Yes, the sugar found in DNA is called deoxyribose sugar. Correct. They're saying that present day distribution of organism, you distribute organism. Let me also give it 40. I'm also waiting for 1.28. 1.28. Family to which humans belong. Hominidae. Hominidae. All right. So I'm waiting. I'm waiting for the answers here. I'm waiting for the answer here and the answer. Yes, here we got it. Homo sapien 1.29, family to which humans belong. No, the family to which humans belong, we call it hominidae. Hominidae. Hominidae, yes. So I'm waiting for now. I'm waiting for which one is it? Hey, I'm still waiting for this one. Which question did I give you? I gave you a food for thought here. The study of fossils, we answered that. Present day 1.2.8. Yeah, 1.2. I'm waiting for the answer. Who has 1.2.8? 1.2.8? I've not yet seen the answer on the. Okay, I think I don't see someone. Okay, we call this one since it is present day distribution of living organisms. It is distribution like different areas, so that's geography, and this is about life. Therefore, it becomes biogeography. Yes, so we call this one biogeography. Yes, correct. So now let's continue. Then we are saying that the further of the future of chimpanzee skull, prognathism, forward position of foramen magnum. Now this one, forward position, it means that you are able to walk on two limbs. Chimpanzees don't do that, so it is only this one, prognathism, meaning that the jaws are protruding jaws. Prognanthus. Similarity between human and apes. Short and wider pelvis? No, because not all of them are able to walk on two limbs. So it's opposable thumb. They can be able to grip. Ne? Like the thumb is opposed to other fingers. Who discovered the fossil called Littlefoot? Who discovered the fossil called Littlefoot? Whom child was Raymond Dart. So Littlefoot was Ron Clark. So it is B only. All right. All right, now let's continue to another question of DNA and then we will wind up with it. Let's go to this question so that you are able now to switch to meiosis. This process, what does it, when you look at it, what does it mean? It means that this process, you see, I said once you see something like this, it's a ribosome, and then what is here? It is a messenger RNA, and whatever you see the boxes or the circles, they can make them circles and then join together, so we call them amino acids. All right, so now we know that this process, this type of we know that this type of process, it is translation. So what is E? It is peptide bond. What is this box? It is amino acid. What is this? It is a tRNA. What is B? This black thing, it is a messenger RNA. What is this? Yes, C is a tRNA. What is D? D is amino acid. What is A? A is a ribosome. Identify the process mentioned above. When you look at the process, we say that it is translation. Correct. Organelle A, we say it is ribosome. Structure B, what is structure B? We say it is messenger RNA. Bond E, we say it is peptide bond. So we know that. Give only letters of molecule that carries amino acids. Letter which carry amino acid. So we said it is C. So we say that here is gonna be C and is copied from DNA. So it means that is B, because remember we said that it contains coded message. Yes, coded message. Bonga Nkonke, yeah, I see you are always participating. Thank you so much. Thank you so much. Sabelo, thank you so much. All right. They're saying that is monomer or building block of protein. Remember, guys, we say that the building blocks of proteins are called amino acids. And the building blocks of nucleic acid or DNA or RNA, we call them nucleotides. So you need to know those two, ne? Yes. So the building block here, it is amino acid. But which letter is it? We come here and then we look at the box, which is D. It can be a box, it can be, yes. Please don't forget about it. Then this question, they're saying that, let's see what are they saying. According to the table below, they're saying how many anticodons. According to the table below, they are looking for the anticodons. When we say the anticodons, we say that no, how many codons? Remember that we say that the codons are those ones which are found where? Just a minute. The codons are those ones which are found on messenger RNA, mRNA. And then what is here? They say it is codon. Also is messenger RNA. Okay, sharp. The thing, according to the table, how many codons code for phenylalanine? I'll look for phenylalanine. That's one. Phenylalanine. That is two. It's this and this. So is that one is fine. What is the anticodon for glutamic acid? Meaning the answer here is 2. Sabero, you can't say that. And they're saying that what is the anticodon for glutamic acid? Glutamic acid. What are they saying? You see, they're saying anticodon. Remember, I asked you, where do we find anticodons? Anticodons are found on what? On tRNA. But what is given here is messenger RNA. Therefore, I have to convert it. What is the glutamic acid? Where is glutamic acid? Is here. Glutamic acid is GAG. So I need to convert it. So it's gonna be G goes with the C, A goes with U, and then G goes with C. Remember we say that G goes with the C, and then A goes with T, in brackets U. So if I have G, it's gonna go with the C. That's why I have C. If I have A, it's gonna go, if I have A, it's gonna go with U, because this is messenger RNA, is messenger RNA versus transfer RNA, meaning that there is no DNA. So I can't say T, so it's U. So correct. And then where there is G, still I'm gonna go with the C. That's why I have G U C. Thank you so much, guys, who have tried. I'm seeing you there giving me correct answers. Zinte. Dakuse. All right. Section of messenger RNA that has the following. Okay, reading from left to right. All right. Let's find out. This DNA-based triplet for the last codon section of messenger RNA. Give the DNA-based triplet. What is there? They say messenger RNA, but they are looking for DNA. So you all must always be keen when you're answering this question. So they said the last triplet, so meaning this. All right. So what am I going to say? I'm going to say that A, 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 A goes with U, because this is mess. They said DNA. A goes with the T, because in DNA we don't have U. C goes with C goes with G, and then C goes with G, meaning that the answer is going to be TGG. The first amino acid coded for by this section of messenger RNA. The first amino acid coded for by this section of messenger RNA. I have to look for it. First amino acid coded for, meaning this one. But they're saying amino acid. But what is here is messenger, meaning that what they have given us here is a messenger. You see? If it is a messenger, what is given in the table? Guys, you must always do that. Not always that you're supposed to convert, and sometimes you have to convert 2 times. So now, if I have the first amino acid for this, remember that they say that this chain is a messenger RNA. But what is on the table? It's messenger RNA. Meaning, do I have to convert? No. So it means that the way it is GAU, the way it is here, it's the same one I'm going to read it from here. So GAU is what? GAU is aspartate. So it means that now this is going to be the answer. Our answer is going to be aspartate. Yes. So why? Because what was given here is messenger RNA, and what is here is messenger RNA. Guys, please don't convert if you see that it's the same, but convert if it is not the same. If this chain was DNA and what is here is messenger RNA, you convert from DNA to messenger. If this chain was DNA and what was given here is transfer RNA, it means that you're gonna convert this into messenger and then convert the messenger into transfer RNA, and then after that you read it from here. I hope you guys, you are getting it right. Yes, I'm seeing some people there that got it wrong, but I think now at least you know it. Thank you. The next question, they're saying that DNA base triplet. DNA base triplet, we are done with that. The first amino acid we said is aspartate. We're done with that. A mutation occurred when which resulted in the following base sequence. All right. Then they're saying that. So you have this chain, but they said it's messenger, correct. Describe the mutation that occurred. They didn't tell us what happened. So it means that the first mark we're gonna get is for you identify which mutation occurred. This and this, they are the same, so it's fine. This and this, they are the same, it's fine. This and this, they are the same, so it's fine. This and this, they are not the same. They are not. This and this, they are the same, so it's fine. This and this, they are the same, therefore it's fine. It means that 1, 2, 3, 4, the fourth triplet, it is the one which was changed. Meaning that, what am I going to say? You're going to tell me that C was replaced by U on the fourth codon. You have to tell us which number was it. You understand? You have to tell us that C, this C was replaced by U on the fourth codon. Then you are able to get what? You told us which number is it? Fourth codon. We'll give a mark, and then you tell us what happened in there. That C was replaced with U. Describe the mutation that occurred. So we are done with that. Explain the effect. Now they want to know what will happen. Now it means that now I'm gonna focus on A, G, C, which was replaced to, it changed to A, G, U. It's the same thing. They say this is messenger, isn't it? So if that is messenger, I will go back and check what is here. It is messenger. Always look at what is given in the table. So since it is a messenger, and also what is given here is messenger, it's messenger, meaning that I don't need to convert anything. You only convert if this and what is on the table here, they don't match. You convert them so that what? You match them. But if you don't do that, you are gone. Therefore, I will find out that C is AGC. What does it code for? AGC. Where is AGC? Is serine. You understand? It code for serine. And then AGU. AGU. AG. Did I say AG? Yes, AGU. AGU code for what? Code for serine. What does it mean? What now? Both of them are serine. So what effect will it be? So now the effect, the last point, I want to see that there will be no effect on the what? On the protein. Meaning that you'll tell us that it code for the same amino acid, therefore there will be no effect on the protein. So this question, they have just split it. In most cases, you bring it as once and you ask you that you explain. And they give you 3 marks. Why do they give you 3 marks instead of 4? Because they tell you that this C was replaced by U. Now you have to tell us, you translate it. After that, you tell me which protein is it. And then, yeah. So it means that, guys, there will be no effect on the protein. And then they're saying that name and describe the process that occurs in the nucleus that results in the formation of messenger. You see, you have to know that. That's what I told you, that you need to know that when they say describe the process, if they say that, you have to know this. Where are you, government? It's here. That formation of messenger RNA, what does it mean? It means transcription. Formation of a polypeptide chain, what does it mean? It means translation. Meaning that now you are able to know that translation means formation of a polypeptide chain. And then now once you say this transcription is a mark, you get a mark. You understand? And then after that, you describe the process of transcription. I think, yes. The last question I'm going to ask you, let me just talk about this. Now this DNA, I'll talk about this. And then after that, I will do what? I will give you a small thing which you need to do for me. Let's go. Identify what is W, weak hydrogen bonds. Guys, weak hydrogen bonds. Don't modify my words. Don't tell me that is hydrogen bonds. This is not physical science. It's weak hydrogen bonds. Simple. What is X? Yes, what is X? It's a nitrogenous base. But which nitrogenous base is it? You can tell us nitrogenous base, or so here is better to say nitrogenous base. Why? Because of, I see a small mistake here. Adenine and thymine, they have 2 bonds, while cytosine and uracil, cytosine and guanine, they have 3 bonds. They swapped them. So nitrogenous base, correct. What is this? Is the oxyribose sugar, and what is this? Is a phosphate group. You have strand 1 and strand 2. I don't know. But this is DNA. So strand 1 and strand 2, there will be still of DNA. All right, let's go to the question and you find out. How many nucleotides are shown there? So you can count them. It's 1, it's 2, it's 3, it's 4. So there are 4 nucleotides. Okay, so we're done with that. They are saying that identify. Okay. Bond Z, we said it's weak hydrogen bond. Give the collective name of X, Y, and Z. Nucleo. nucleotide. Yes, it's a nucleotide. So why? Because remember, nucleotide is made up of a phosphate, a sugar, and a nitrogenous base. Okay. They're saying that state the natural shape of DNA. We talked about it. It's double helix. Guys, don't modify. I don't want modification there. Don't say double stranded is wrong. Double helix is wrong. It's double helix. There's nothing else. Don't modify words. You want to modify words so that you are benefited or you are protected. Your answer is correct when it's wrong. Name two phases, name two places in animal cell where DNA is located. We answered this question. We say that the first one you can talk about where DNA in animal cell, nucleus, then mitochondrion or mitochondria. Yes, so those are the two. All right. So that's it. Simple like that. Simple like that. Uh-huh. I think that's it. I think that's it. Let me do this one and then we go to the last. They're saying that what is X? What is X is deoxyribose, because they say this DNA, they say this RNA is deoxyribose sugar. You can't say sugar because they say this DNA. If this is adenine, then this is the thymine. Are you sure? No, because this is messenger RNA. So if adenine goes with uracil, because RNA doesn't have thymine. You understand? So it is replaced by uracil. So this is uracil. Then they are saying that name the part of the cell where this process occurs. You see that I'm converting DNA into messenger RNA. Therefore, they're saying name the part where this process occurs in the nucleus, meaning that where is transcription takes place, because that is transcription, and they have already also talked about it, that is transcription. And then we got the next one. They're saying that tabulate two differences between transcription and DNA replication. Between transcription and DNA replication. They're saying that transcription and DNA they want you to give us the difference between DNA replication and transcription. Nucleolus. Not nucleolus. This process doesn't happen, it doesn't take place in nucleolus. It takes place in the nucleus, not nucleolus. All right. So the answer is gonna be you tabulate. The first thing, remember, I told you tabulate. Tabulate. When you tabulate, you say DNA replication, and then here you say transcription. Then you give us the differences. But they did they say structure? They just say give it the differences, meaning that it doesn't matter. Give us two. Here, at the end, you form DNA. Two new DNA are formed, meaning that it results in the formation of DNA molecule. Here at the end, it results in the formation of messenger. All right, good. Here, two strands act as a template. Here, one strand acts as what? A template. So they want two. So you can also look for others. Yes, you can also talk about here in the process of DNA replication, DNA nucleotides are being used. Here, RNA nucleotides are being used in the process. You understand? So you can talk about here. It involves adenine, guanine, cytosine, and thymine. Here involves what? The new strand. It will have adenine, guanine, uracil, and cytosine, meaning that there is no thymine in the strand which is formed. So you can make sure that here, when they say tabulate 2, we mark the first 2. If you give the answer, each strand serves as a template. The other one strand serves as a template. Yes, correct. Ntombi, correct. Thank you. All right. So I'm going to do this one. I was going to skip it, but let me just do it. Last. Yes. Yes. Let me just do it. They're saying that a mutation occurred on the messenger RNA as shown below. All right. Name the type of mutation because this thing is happening on the gene, so it becomes gene mutation. So it's a point, but which mutation happened? Let me check. They are fine. So it happened here. This the same. So it's 1, 2, 3. 1, 2, 3, 4. Oh, it happened on the 4th. Still on the 4th. Give a reason for your answer because the nitrogenous base C was replaced with U. You see? Yes. Nitrogenous base C was replaced with U. Yeah, sometimes you can also call it point mutation. The table below shows. Let's go back to this table, guys. They're saying that the table below shows messenger. You see? State the number of different amino acid coded for by the original sequence of messenger. Here, how do I answer this? Okay. State the number of different amino acids coded for by the original. You see this word? It's a key point. State the number of different amino acid coded for by the original sequence. They said original, this one. Now I need to find out. The original sequence, it's what I need to know. Remember, they said it's messenger. And remember here, they also said it's messenger. Meaning that this thing is messenger. Do I need to convert? Nope. I don't need to convert. So I'm going to have. So I need to find out what is this, 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 what is this. The one which repeat itself, I don't count it. So it's going to be AUG, AUG is methionine, meaning that one is gone. Saying GAA, GAA, GAA is glutamic acid. So that one is gone. Those are two. AUA, isoleucine. So those are three. That one is gone. CCG is proline. That is 4. That one is also gone. CCA is proline. You see? Now this is repeated. So this is counted as 1. So I have 1, I have 2, I have 3, I have 4. Then I have GGA. GGA is glycine. It is another one because this one is the same. They didn't say that state the number of amino acids coded. They said different. So the one which is the same, I will pass it. Why? Because it is repeating. Because they said different. It is not different. Therefore, I'm going to say 1, 2, 3, 4, 5. So the answer becomes 5 amino acids. We got the next one. Give the anticodon on the tRNA molecule that carries the amino acid isoleucine. They want transfer RNA. But what is this? Let me first clear it up. Isoleucine. This one. But they want the transfer RNA, meaning that I have to convert because this is messenger. I'm going to say AUA is going to be converted into transfer RNA. It's going to be U AU. So that becomes the what? That becomes the answer. Uh-huh. Then we go to the next question. Use the information in the table to describe the effect of mutation in the protein. So now I need to find out. We said it was on the 5th, ne? Where is it? It was this. Now I need to know what does this code for and what does it code for. But don't forget that this is messenger and this is messenger. So I don't need to change anything. I don't need to change anything. So it's gonna be CCG and then CUG. I'm not changing anything because it's the same thing. This and this. Yes. So it's gonna be name the table. Use the information in the table to describe. So I need to find out what does this code for. CCG. CCG is proline and CCUG. CUG is leucine. You see now this one is different. So you're gonna tell us that CCG was changed to this, ne? CUG. You understand? Yes. It was changed to that, meaning that did they talk about, yes, on the fourth codon. You understand? Yes. And then now this CCG code for proline, while CUG code for leucine. So therefore there will be a difference, there will be a different protein which will be formed because of the difference in the amino acid which are being coded for. So you have to mention that this one will code for this one will code for this, and also tell us that this G, is it G? No, this C was changed to U on the fourth codon. Yeah, it was the fourth, ne? Yes, 1 2 3 4. Yes, on the fourth codon. So you see now we are able to get this. Oh, by the way, thank you so much, guys, to those people who are subscribing to our channel and those people who like our videos, those people who share our videos, those people who use our distinction material books. Yes, and all those who support in any way. I don't know which, any way it could be, just to talk that, oh, do you know this channel called Thunder Duke? Yeah, they always talk about the predictions and all the predictions are always correct. All right. I think I'm done with DNA. I'm done with DNA. The only thing which remained with DNA is just to remind you about DNA profiling. This one is simple. You have to know it. And then another thing is basic calculation. So I'm going to just talk about the basic calculation and then we continue. Let me use the distinction material book. Maybe I will just use a question from there. Did I leave any question on the basic calculation? I think so. Yes, basic calculation. I'm talking about that. If DNA has 5,000 bases, 500 is guanine. What is the ratio of thymine to guanine? To cytosine. All right. If DNA has 5,000 bases, 5,000 bases are for DNA. DNA has 5,000 bases, and then 500 bases are for what? They said 500 bases are for guanine. 500 bases are for guanine. What is the ratio of thymine to guanine? So whenever they ask you this question, you have to first tell us the story that A goes with T, C goes with G. You must always know that if this one is 500, then definitely this one is 500. So what is the So when I combine the two, I get 1,000. But remember, the total is 5,000. Sometimes they can bring it in percentages, whereby now you subtract from the percentage. Maybe if this is 20, the other one is also 20. So 20 plus 20 is 40. Then you subtract. So it means that if I get 5,000, I minus it 1,000, then I'm going to remain with the 4,000. So the 4,000 is for both this and this. Therefore, divide by 2 because they're equal. So it's going to be 2,000. So it means that this is 2,000. This is also 2,000. But I need a ratio of thymine to guanine. So add thymine to guanine. Thymine, if you come here, is 2,000. It's 2,000. We see it here, and guanine is also is 500. You see, it's 500. So once I know that, so I'm gonna divide it with a number which is make sure that it's divisible for both. So it's 500. It can be 500, the number which is here. It can be a number which is not there. So divide by 500. Sorry, 500. So this one's gonna be 4, and then this is gonna be 1. So the ratio is gonna be 4 to 1. Guys, don't rush when you see such questions, because the first objective they can say 1 to 4. Because when you solve the 4 and you saw the 1, you rush. Someone was giving me bonga konke. Yes, thank you so much. Your answer is correct. So please at least know before you go to the paper, at least make sure that at least you know them. Ne? Yes, thank you so much. Then we go to the next question. Meiosis. 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 Yes, what is meiosis? Yes, this is a food for thought. Food for thought. What is this? This is the food for thought. What is this? V. What is V? But remember, this one was obtained from this side. Therefore, V, if this is thymine, therefore V is going to be adenine. What is molecule? It's nucleotide. What is this molecule? DNA molecule. Which process? This is DNA replication. What is this bond? Is weak hydrogen bonds. So you see, so we're able to answer all these molecules. View, we done that. This Q, we said it's nucleotide. Bond Z, we said is weak hydrogen bond. V, where is V? Or V, we said is adenine. Where in the cell does this takes place? It takes place where? In the cell. It takes place in the nucleus. Name the phase of cell cycle where replication takes place. Name the phase that is interphase. Remember, we said that DNA takes place before the cell divides or during interphase. I think we are done with that. Let's go to meiosis, and then we smash some questions about meiosis. Right. Meiosis. So in meiosis, guys, it's simple. Meiosis is very simple. Very simple to pass it. Tell me the functions of meiosis. Meiosis is very important for bringing about variation. Number 2, to form haploid gametes. Number 3, to maintain the chromosome number from generation to generation. So now we know the function of meiosis. Where does it occur in plants? It occur in animals. It occurs in testes and the ovary, and then in to produce sperms and the ova in animals. Then the ovary to produce ovules in plants, and the anthers produce pollen grains. That is in plants, right? Then we go in other part. Then we can talk about what is a karyotype. A karyotype is you are trying to describe the diploid state of a cell, showing the arrangement, the number the arrangement, the number, the size of the chromosomes which are found in that cell. It must be a diploid cell. Yes. So that's why we call it a karyotype. Karyo means a nucleus, which means what? Which means which nucleus, which type of nucleus does the cell have? Then when you need to talk about crossing over process where the genetic material is being exchanged between two chromatids of homologous chromosomes at a point called chiasma. You need to know that the crossing over takes place it takes place during prophase 1 only, prophase 1, and then that point we said is called what? Chiasma. The reason why crossing over is supposed to take place to bring about genetic variation. That's why I always ask you a question. Are you sure? Your father is your father. Yeah, because of variation. Even if you don't resemble him, still it can be possible that is your father. Anyway, I always tell people that if your mother is still alive, watch out. Anyway, I didn't say anything, guys. The next question is you need to know the different phases. What happens in prophase? The major phase which happens in prophase is prophase 1. You need to know that prophase 1, what happens there, which differentiates from other phases, is the crossing over. What happens in Mera phase? Mera means middle. Mera phase, middle, which means the chromosomes are arranged along the equator. You understand? In homologous pair, randomly. That word randomly, homologous pair, we want to see it. Then in anaphase, these chromosomes, they move to opposite poles. Chromosome, not chromatids. In prophase, in anaphase 1, ana means apart. Anaphase. You will see that the chromosomes, they are apart. You understand? Yes. Then telophase means they reach the terminal. Yes. Meaning that now the chromosomes have reached the terminal. So in prophase 2, what you need to know that we talk about chromosomes and chromatids. We don't talk about the word homologous. We don't talk about that. Yes. We only talk about chromatid in anaphase 2. Don't talk about anything related to chromatid when it's not anaphase 2. We only talk about chromatids in anaphase 2. So they can ask you the difference between meiosis 1 and meiosis 2. Then there you have to tell us that, look at phase by phase, that in meiosis 1, in anaphase 1, yes, there is crossing over. Anaphase 2, no crossing over. In metaphase 1, you have random arrangement of chromosomes. Yes, okay. Chromosomes, homologous chromosomes arrange along the equator randomly. Homologous chromosomes. There you say that chromosomes align along the equator singly or individually. We go to anaphase, you say that chromosomes move to opposite poles, are being put to opposite poles. Therefore, in the other side of anaphase 2, you talk about chromatids move to opposite poles. So you see that you are able to look at phase by phase, then you'll be able to interpret these questions. So, and then another thing which you need to know that non-disjunction. Non-disjunction is the failure of the chromosomes to separate during metaphase. You see? Yes. Did I say metaphase? No, man. During anaphase. It can be anaphase 1, it can be anaphase 2. Then, what does it result to? It results in Down syndrome. So you need to know that. Then, you see now you have all the questions. Then you need to know the structure of a cell. When they talk about the variation, you need to look at the structure of the cell. Look at the structure of the cell before and after. If there are variations, make sure that those variations, you take them correctly. Label everything which you can, whether you know it or you don't know it. If you see that you suspect that this is called this, call it. So that when you are marking, at least we look for those 2 marks. Yes, you don't lose if they ask you to draw. Don't forget the caption. I repeat, don't forget the caption. Don't forget the title if you don't understand the meaning of caption. All right. Then you need to also to to know the number of chromosomes. What happens, the number of chromosomes in meiosis 1. In meiosis 1, the number of chromosomes, they are still diploid, except in telophase 1. They are diploids, 2 sets. And then in meiosis 2, they are haploids. Listen, if the question is not mentioning that this is a human cell, don't talk about 46 chromosomes. Look at the cells there. If they give you the, for example, let me just give you an example of this. They ask you a question. How many chromosomes do you see there? You tell me 46. Do you see 46? Do you have eyes? Make sure that you talk about what you see. How many chromosomes? 1, 2, 3, 4, which is chromosomes. Simple. Don't talk about 46 if they have not mentioned a human cell. We don't say that this is meiosis for human. No. We say meiosis, but we just use human because we are humans. But sometimes the question, in most cases, not even sometimes, if they want to talk about human, you will see the word human in the question. That human, this is a human. Then they will say that, how many cells would there be if it was a human cell? How, if this is, you see that it is the crossing over is taking place, then they would have said that, how many chromosomes would there have been if it was a human cell in the same phase? Then that's when you say that, oh, this is 46 because it is meiosis 1. But if they ask you how many chromosomes do you see here, you tell us it's 1, 2, 3, 4. How many chromosomes will there be at the end of meiosis? Of this process. Don't tell me it's 23. They didn't say it's a human cell. These are 4 chromosomes, meaning that at the end of meiosis, it will be 2 to 2. Yes. So, meiosis is simple. The problem is we just panic. And the moment you panic, you lose it. Please, don't let me down. I want marks. When I see that you get marks, I feel happy. Yes. Susan, I hope you hear me. And Love, I hope you hear me. Nomfundo, I hope you hear me. Tati, I hope you hear me. Winnie, I hope you hear me. Zintle, I hope you hear me. Georgie, I hope you hear me. Lole, everyone who is there, Bonga, I hope you hear me. Zinte, I hope you hear me. There is Zintle and there is Zinte. Okay. You guys, I hope you guys will hear me. Please don't let me down. Make me proud when I see that at the end of the day, you get the answers right. So guys, as usual, eh, it's always, you're gonna do this question for me. I'm going to come back. Why? Because it's time for prayer. It's Magharibi prayer. So I'm gonna give you, I can't pray for 5 minutes. I'm gonna give you 10 minutes. Do this question for me. 10 minutes. I'm going for prayers. Prayer time. So if it's prayer time, there is nothing I can do. I have to go. You understand? Yes. So I'm going to also go and pray for you the same way I prayed for you in paper 1. So please, guys, do that for me. When I come back, we smash it. Thank you so much. Hmm. All right, guys. Let's continue. Let's continue from where we stopped. Someone saying it's no sound. Yes, I was not yet. I had not unmuted you guys, but now it's fine. Okay, no sound, no sound. The sound is there. Okay. All right. So the question was saying that you need to, we need to, answer those questions. Let me see. Answered them. They're saying that what is A? So we need to label everything. Remember, we need to first label everything. What is A? Let me see what people said. Oh, yeah, a lot of answers here. What is A? A is a cell membrane. Okay, correct. B is a spindle fiber. Correct. C is a... where's C? Didn't write C. Okay, C is a chromosome. All right, C is a chromosome. So, give the observable reason for your answer. Okay, identify the phase of meiosis shown above. This is, remember, these chromosomes are single. So when they are single, this is supposed to be on the sides so that you're able, it must be always on the horizontal scale so that you are able to, if you see them that these spindles, they are moving like from here to here, they are moving up and down, it means that you have to turn them so that they look like this, so that you see that, okay, they're either in the middle or they are being separated. So we are saying that which phase is this? This is the chromosomes are in the middle. Remember ipmed, which means interphase, prophase, metaphase, anaphase. Yes, I hear you. Anaphase, and then telophase. So that's what it means. So now it means that you have still, it can mean in between, preparation phase, middle, meaning the chromosomes are in the middle, the chromosomes have been pulled apart and chromosomes have reached the end or the terminal. Yes. The chromosomes have reached the what? The terminal. So that is it. So how can I answer this? Identify the phase. So it is metaphase, because the chromosomes, metaphase, the chromosomes are in the middle, but they are single. So if they are single, then it means that it's metaphase 2. Give the observable reason for your answer, because chromosomes are along the equator singly, or individual chromosomes are along the what? The equator. Then they are saying that identify structure A. A is cell membrane. Yes, we said it. B is spindle fibers. Describe the role of B in the movement of chromosomes. I saw someone answering that question. It pulls the chromosomes or chromatids in opposite direction. To the poles. It's better to say that it pulls the chromosomes or chromatids to opposite poles. Yes. Thank you. Someone is saying that there is no sound. Serious. There's no sound at people who are online. Let me ask them properly. Someone saying they don't hear me. Hey, serious? Let me check and see. So you guys, you don't hear me, ne? Okay. Someone saying yeah, the sound is there when guys stop it. The person who can't hear me, make sure that you unmute yourself. I mean those ones who don't hear me. Maybe you are still at the back stage. So play the video to the next, yeah, so that is live, not playing maybe after 10 before 10 minutes, something like that. All right, we continue, guys. Mm, thank you so much, those people who have updated me. uh They're saying that identify structure A, we are done. Describe is done. Draw the diagram. Draw the diagram. Draw a labeled diagram of structures as it would have appeared in the final phase of meiotic division, showing the correct shading. So guys, whenever they ask you to draw, make sure that you draw the right thing. Here they say the final stage, how it would have appeared in the final stage of this meiotic division, meaning that the final stage of meiotic division is telophase 2. Telophase 2. That's what it means. So telophase 2. So now it means that I'm going to have to draw. Whenever they ask you to draw, don't forget the caption. Caption number 1, you get a mark. Don't forget to label at least 2 things, but don't label 2 because you might label wrong things. At least label 4 if you could, then label 4 so that you give you 2 marks. So it means that now you have 3 marks out of 4, and then draw the right thing. So it means that if you look at it, I'm gonna draw it, yes, but now if I split it like this, I make sure that I split it right. Remember, in telophase you have the nucleus. The nucleus is back. So, and then this shade, if I use the one which has a shade, it must show the what? The shade. Then if I use this one, the second one, meaning that one, it will have a shade up and a shade down. You see? Or you draw those ones without a shade. You draw those one without a what? A shade. Without a shade. Even the other one has no what? Has no shade. But you draw proper things. Don't draw like me. Hey boy, hey boy, hey boy, hey boy. Then you label as you tell us this is a cell membrane, this is the nuclear membrane, this is a chromosomes, this is a centromere. So you label it. You label it. You label it. Ne? Yes. Make sure that what? You label it properly. Then you'll be able to get to what? The marks. Yeah. So we got the next question. Next question, let me check for you this question. It's a question which I wanted you to do. Yes, it's page 13. Let's go to page 13. Page 13, page 13, page 13, page 13. Yeah, page 13, page 13. Yeah, there we go. Let me first do the last one before I come to page 13. I do the last one before I go to page 13. Yes. What is A? So you look at this structure, at this thing, and then you tell me. What is A? So this is homologous chromosome. What is B? This is centrioles. What is C? It is spindle fibers. What is D? This is homologous chromosomes, but they have variations. This variation, maybe they're going to ask you what are the what caused those variation. It is crossing over which makes those variation. But remember, I told you that now once you reach to, if they ask you identify the phases, when you reach to this one, ne, I told you that these spindles are supposed to be in the horizontal way so that you're able to identify it properly. Let me show you what I was trying to mean. So when you get something like this on the paper, make sure that you rotate it like that. So you see that now the spindles, are on the sides. You see, they are all. So you see now you are able to identify that these chromosomes are in the middle. So this is metaphase. Metaphase. But metaphase what? It is metaphase what? It is metaphase 2. Metaphase 1 because they are 2. See, they are still in pairs. And if you look at this one, check this one and check this one. They are still on the sides. So what is happening to them? But remember, there is this action which is happening. This action only happens in prophase 1. We don't have that action anywhere else except in prophase 1. So it means that now I'm able to identify which phase is it, and then I'm able to identify the labeling of those Z. So make sure that you guys, please take note of this, that from metaphase and anaphase, yes, we don't have nuclear membrane. So you can't say that this is the nuclear membrane. This is a cell membrane, ne? Or plasma membrane. This one, it is prophase. Prophase, you will see the nuclear membrane is starting to what? To disappear. That's why they are dotted. This one is a cell membrane. This is the nuclear membrane. You see? But in metaphase, we don't have a nuclear membrane. We only have the cell membrane. So I think that is clear. So now identify the phase in the diagram above. So we know that the phase is which diagram 1. So it is because of the crossing over. So it is prophase 1. Prophase 1. Then give 3 reasons for your answer. Eh? Oh, they want 3 reasons. Why do you say it's prophase 1? First of all, we can see that. What do you see? We see that the nuclear membrane is disappearing. So that's number 1. That the nuclear membrane are disappearing. Yes, you see that the chromosomes are homologous in the nucleus, whereby they have what you call crossing over. Yes, they are crossing over, meaning that we're able to see that, oh, this is what? This is the prophase 1. Prophase 1 crossing over only happens in prophase 1. Number 3, we see that the spindles are starting to form. Spindles, there is formation of the spindle fibers. The spindle fibers are starting to what? To form, to generate. Therefore, it shows that it is prophase 1. Yes. They are saying that describe the process that takes place at A. Here you have to describe the process of crossing over. Chromosome of homologous chromosomes, they align together. Then the chromatids of the homologous chromosome cross over at a point called what? Chiasma. The genetic material is exchanged, then they split so that they have genetically different what? Combination of genes. So you have to describe the process of crossing over. Please don't go to the paper without knowing the process, the process of crossing over. Then they are saying that identify the phase. So basically, those people are using the genetic. I opened when it's just the. Okay, it just took us direct to what we are asking. All right, so this is what I'm talking about. That please make sure that homologous chromosome align together. One chromatid of each homologous chromosome overlap with its homologous partner. Crossing over takes place at a point called chiasma. Yes, the chromosomes separate in such a way that each has new genetic material from both chromosomes. So you tell us it's from prophase 1, you tell us the homologous chromosomes aligning together, you tell us that they cross over at a point called chiasma, you tell us that they have the new genetic information which has been exchanged, meaning that material. So we need to know those key points before you go to the next question. So they're saying that describe okay, we're done. Describe the result at the end of meiosis if chromosome D failed to separate. So chromosome D, so it means that it is gonna be what? It's gonna be non-disjunction. So you're gonna tell us that it is called non-disjunction. Describe the result at the end of meiosis if, yes, so if it is non-disjunction, so it's gonna result in one cell having now an extra chromosome, and then another one is going to have short of a chromosome. But now, did I say it's a human cell? So do I have to say it's 23 chromosomes? They're not talking about Down syndrome, so they're just talking about non-disjunction. All right. So what does it mean? It means that now if they separate, normal separation is supposed to be like this. Whereby this one comes this side, this one goes that side. But now, meaning that each cell is supposed to have 1, 2, 3, 4. It must have 4 chromosomes. Even this one is supposed to have 4 chromosomes. Why don't I say 23? Because they didn't talk about human cell here. Then now, if you look at it, you say that if all of it goes to one side, it means that I'm going to result in a cell having 1, 2, 3, 4. 1, 2, 3. Now this one is going that side, meaning it's going to have 3, and then this one is going to have, now this one is going to have 1, 1, 2, 3, 4, 5, because the whole thing went to one side. So it's going to have 1, 2, 3, 4, 5, meaning that instead of having 4, it's going to have 3, and instead of having 4, it's going to have 5, meaning that it will have the cells, one cell will have an extra chromosome, while another one will have, it will have less of a chromosome. Yes, so that is non-disjunction. And if this is a human cell and this fertilizes a normal gamete, then it means that it's gonna result in an individual having 47 chromosomes instead of 46. You understand? So if it was a human cell, and it is called Down syndrome as the condition. So describe the process of DNA replication. Now this one, we have discussed about DNA replication. Yes, so determine the sequence. So we did DNA replication. We did it very well. Yeah, very well. So remember, we talked about this is messenger, this is what is this is showing. So it's one strand. DNA is says strand, but now I need to find out where did this strand, so it might be the template A U C A U C. A goes with the T, meaning that this strand S was the template, because if you look at this is complementary to that, meaning that the image of this was there. After that it left. So this is a nucleotide. Did I say nucleotide? This is amino acid. This is transferrin A. This base is anticodon. This is messenger RNA. The base on the messenger is called codon, and then this is a ribosome. So we discussed this. So I think, guys, you are very good in that. Let me look for the next question of meiosis. They can ask you a question here. How many chromosomes? How many chromosomes? How many chromosomes? How many chromosomes will there be? How many chromosomes will there be at the end of meiosis for this process? So it means that here they're gonna be 1, 2, 3, 4, 5, 6, 7, 8. Meaning that at the end, remember, it is haploid. So it's gonna be 4. So 1, 2, 3, 4, 5, 6, 7, 8. How many chromosomes if it was a human cell? Then you would have said 23. You see? Yes. We go to the last question of meiosis, just to be as a food for thought. Okay, it's page 26 and the page 13. Page 26 and the page 13. Page 26 and the page 13. Okay, it's page 26, page 13. Okay, we need to talk about this question of a karyotype. So I'm going to talk about karyotype. Let me just go to page 28. Let's go to page 28. Sharp. We are saying that, look at the karyotype. The question comes as a karyotype. How do I know? Describe the structure of a karyotype. A karyotype is made up of 46 chromosomes of a human karyotype. Yes, they always talk about the karyotype you talk about here in most cases, a human karyotype. So it's made up of 46 chromosomes from chromosome number 1 to pair number 1 to pair number 20 are called autosomes. The 23rd pair is called gonosomes. The gonosome has X and Y. X is bigger than Y or is longer than Y, or yes, the X. If an individual has X and X is a female. If an individual has X and Y is a male. So you see. Yes, so you describe it. Yeah, a karyotype. But they can bring a question like this and ask you, is this a male or a female? Identify which one is a male, which one is a female. So you have to know which one is a male, which one is a female. So in this case, it's gonna be, this one is gonna be a male, while this one is gonna be a female, because remember we said that X is bigger than X is bigger than what? Y. So it means that if this is X, then this is Y. If this is X, that is Y. This is X. You see that these ones, they have the same size, meaning that is X and X, or this is X and Y, because we say that Y is shorter than the what? Than the X. So that is it. So let's go and then find out what are they saying in this they're saying that name the specific type of chromosome numbered 1 to 22. We said is autosomes. They're saying that each pair or shown in the homologous pair, okay, state the origin of each chromosome in the homologous during the zygote formation. Meaning that one came from the father, the sperm of the father, and one chromosome came from the father, and one chromosome came from the mother. That is the mother's what? Ovum, and then the father's sperm. Then they're saying that list 3 characteristics that chromosome in the homologous pair have in common. We say that they have the same shape. Yes, the same shape, the same size. Yes, same location of genes, not same genes, same location. For example, if the gene for example for big eyes is here, even on the other chromosome it will be on the same spot, but this one could be small eyes, and then this could be big eyes. You understand? But it might not be the same gene, same genetic composition, but it's the same type of gene. They will be found at the same locus, same location. This is what I say. This is what I mean. That these chromosomes they are arranged in pairs, pairs in different. They have the same shape, same size. You see, the same genetic composition in terms of the location. You understand? Like if you have the chromosome for gender at that point, even the other point it will be the same. If you have chromosome for long hair at the same point, so like that. Yes. Yes, Jeannie. Yes. That's what I was trying to explain. Explain one observable reason why the two individuals are not identical twins. Identical twins come from a same sperm. So if they come from the same sperm, meaning that the sperm only has one chromosome. Either is X or it is Y. So if it's the same sperm which fertilizes the ovum, they must be of the same gender. These ones, one is a male, one is a female. So they can't be identical twins. Yes, they can't be identical twins. So what are they saying? Explaining one observable reason why the two individuals are not identical twins. Then the remarks. Then it means that you have to tell us that the chromosome pair number 23, yes, of individual one has X and Y, while the chromosome pair, yes, number 23 for individual 2 has X and X, which means that they were fertilized by different sperms. Yes. Describe non-disjunction that may lead to Down syndrome. So now here you have to tell us how does non-disjunction leads to Down syndrome, whereby you have chromosomes. For example, I'm gonna just draw something like that. There are two. Let me just say two. They fail to separate. Okay, let me make them three. It's the same thing. Let me make them three. Now they fail to separate. Now you're gonna have here, meaning that they are pulled to opposite poles. They are pulled to opposite poles. You see, that's normal. And then at the end of the day, you form 1, 2, 3. And then also here, 1, 2, 3. So, but when non-disjunction occurs, the whole chromosome will be pulled to one side. Meaning that I'm going to have a cell which have 1, 2, 3. You see? It's being pulled here. This one is going to be having 1, 2, you see? And then the third one is going to have nothing because the whole chromosome is pulled to that side. So it's going to result in 1, 2, 3, 4. 1, 2, 3, 4. And then this one is going to have 1, 2. The second one is going to be 1, 2. That's what I've been explaining, meaning that now this one is going to have 3 chromosomes. It's going to have 2 chromosomes of the same kind. All of it went there. And then this one is going to have nothing. It's going to have 0. Meaning that if this one fertilizes the ovum, the normal ovum, if this was a sperm, yes, that sperm is going to also have normal fertilization, meaning that if this was the sperm, the normal sperm, so this one is going to combine with this, meaning that now this chromosome, they are going to be 3. They are going to be 3 in that individual. If it is on chromosome number 23, 21, then we're going to link it to trisomy 21. Since there are 3, it is trisome. Some is a body. So it's describing a body which has 3 chromosomes of the same kind. But if it is 21, it happened on chromosome number 21. So it becomes trisomy 21. And that description is what you call Down syndrome. So that's how you're supposed to explain. But you know it, ne? Let me just show you based on here. Yes, each nucleus, non-disjunction is the failure of the chromosome to separate. So each nucleus should contain 23 chromosomes after meiosis, but non-disjunction occur. Okay, this is where the answer is. You tell us that one nucleus will contain 22, while another will have 24. Yes, when either of these, yes, fuse with the normal gamete, which is 23, it will result in 45 or in 47. You understand? The chromosome, instead of what? 46. So we know that. So this may lead to Down syndrome. How? Yes. So the child will have 44, 45 autosomes, meaning that plus 2 from the sex chromosome. You understand? Making them 47 instead of the normal pair. Meaning that you instead of 46, you'll have 47 instead of what? 46. And then because it happened on chromosome number 20, check. with 21 chromosome, with chromosome number 21, meaning that at chromosome number 21, there are 3 of them instead of 2, because they are supposed to be 2, one from the father, one from the mother. But now the other time, the father brought 2 and the mother brought 1. You see? So it resulted in that chromosome being 3 of them. So instead of the normal, which is supposed to be 2, and then Down syndrome is going to be yes. So now that is it. Oh, someone is saying, oh, sorry, I didn't see the answer. I didn't see the question. What what is a somatic cell? A somatic cell is what called a body cell. These are cells of the body other than the sex cells. Yes. Uh-huh. That is it. So that is it, guys. On we need to go to genetics. We need to go to genetics. We need to go to genetics. Let's just check here, page 34. Yeah, 34. Cool. Yes. 34. Aha. Let me just finish up with that, and then we shall say that thank you so much for what? For okay. Yes, page 34, page 26. Okay. They're saying how many autosomes are shown. Autosomes, remember, these are chromosomes which are not sex chromosomes. So in this case, they didn't say. Did they say the diagram below shows part of abnormal human karyotype? They're saying how many autosomes are shown. Shown, not how many autosomes are in this human. Autosomes. So it's gonna be 1, 2, 3, 4, 5. So there are 5. Remember, this is a sex cell, sex chromosome. Name the type of chromosomes represented by these are gonosomes. Gonosomes. Gonosome. Yes. They're saying that this order represented, it is 1, 2, 3, meaning that I have 3. Trisome. Is tri means 3, zome means a body. So trisomy 21. Oh, Down syndrome. And process during anaphase of meiosis that resulted in this. So it is, we say that is non-disjunction. Non-disjunction. State the gender of this person. One chromosome is long, one chromosome is short. So it is a male. I think now you're able to answer those. Then page 26. Yes. Page 20. Yes, this page. Let's look at this. Page 26. They're saying that what is A? We say that these are where the this process is crossing over. So it means that P is chiasma. And then what is P? It's centromere. What is A? It's a chromatid, showing just one chromatid. They're saying that the organ in humans where meiosis occurs in the testes. Process represented above, crossing over. Identify what is P. We say it's chiasma. What is Q? That is centromere. And what is R? That is chromatid. Done. Genetics. Genetics. So let's go to genetics. We are going to what? Genetics. Genetics. No question? No question? No question. Okay, let me see which genetic question I'm gonna start with for you. Or I should give you, think I should give you a breakfast. Maybe breakfast means some terminologies. Any break, we call it breakfast. Breakfast means, breakfast to me means terminologies. Yeah, it means terminologies. Okay. Let's start. Should I start with the dihybrid? You hear it, you feel it. Yeah, let's start with the page 23. I'm gonna start with the page 20. Let me start with the page 23. There we go. Yes. What is that? That is genetics. Guys, don't forget that in genetics, there are some things which you need to know. Make sure that you know how to do a genetic cross. So if it comes to genetic cross, make sure that you know them very well. Don't lose those marks for the genetic cross. Yes, let me just do a small recap. Don't forget that you start with the P1, F1, P1, F1. If it is the second generation, you say P2, F2. Don't forget this. In most cases, kids write P1, they forget F1. We cross. You lose a mark. This is a free mark. Don't forget to write these. Memorize them. If it requires you to take an energy drink to memorize this, memorize it so that
recording stopped.
memorize it so that you are able to get it right. You understand? Yes. So in this case, I will say that P1, P1, F1. So we give you a mark. When you say meiosis and fertilization, we give you another mark. The phenotype is always spoken in the what? In the question. Then the genotype, you get it from the phenotype, depending on which one is dominant, which one is recessive. Yes. So please, 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 you need to know that. Then you need to know you can either use a Punnett square method or you can use just a genetic just crosses. So you can use that, or you can just use these crosses like this. You can just cross them like that, and make sure that when you are using the cross, I prefer people using genetic Punnett square. Why? Because this like this, people end up writing them like this. And then they write T, 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 T. You ask yourself, what is this? What is this? Yet you have messed them up like that. You lose marks. So if you can't do that, just use the Punnett square. Okay, let's look at some few questions. This question, okay, I think that's it. There are some questions which are going to come. Let me just do a small what? A small recap. When it comes to genetics, you need to know some of the terminologies. What is a locus? What is a gene? What is a chromatin network? So those terminologies, you need to know them. Yes, these ones. Chromatin network, chromosome, thread-like structures, gene. These are a portion of DNA that control a particular character. Alleles, alternative form of a gene. Dominant allele, that is a gene a gene which is expressed. Allele which is expressed in a heterozygous state. Recessive only expresses itself in a homozygous state. You need to know this. Phenotype, the physical appearance of an organism. Genotype, the genetic composition of an organism. Homozygous means that the letters are the same, meaning that they have the same kind of allele. If they are dominant, they are dominant. If they are recessive, they are recessive. Heterozygous, it means that they are different. Capital T, small t, or capital B, small b. They are heterozygous. So you need to know those terminologies. Monohybrid means a genetic cross which involve one character. Complete dominancy means that one allele is completely dominant over the other. When do I see this? You see that whenever you do a genetic cross whereby involves one is a capital letter, one is a small letter, yes, that is it. It means that it's complete dominance. One is completely dominant over another allele. So in complete dominance meaning that neither of the allele is dominant over the other. For example, if it is white crossed with black, and then you produce gray. You produce gray, meaning that neither of the white nor the black is dominant over the other. Both of them they have the same power. So you need to know that. Meaning that if both of them are dominant, it means that white is going to be capital W, black is going to be capital W. Meaning that gray will be, capital W, capital B. You understand? Yes. So it is an intermediate. So in this case, you have to look for the word an intermediate. Codominance means together. They both alleles are being expressed in the phenotype. So you need to know those things. I'm just doing a highlight. Yes. So you're able to see it. This is codominance, meaning that red and blue, you see them that you have them mixed together. Then you have here red and white, you've produced pink. That is incomplete dominancy. And then in complete dominancy, you have red and white, you produce red, meaning that red is completely dominant over white. Then we go to the next question. Then we got the next highlight. Here, sex determination. Here, whenever you talk about sex determination, basically you have to tell us that males, humans have 46 chromosomes. Basically, you are describing a karyotype in this regard. It's like a karyotype. Yes, you see that you only find out that you are describing something like a karyotype. It's not a karyotype. So the 46 chromosomes in a human. 1 to 23 is gonozome, sorry, is orosome, 23rd is gonozome, X, if a person have X and X is a female, X and Y is a male, you understand? Meaning that if a sperm having X fertilizes the ovum having X, the individual is going to be XX, meaning that it's a female. If a sperm having Y fertilizes the ovum which has X, it's going to be XY, meaning that it's a male. So there are 50 chance of a person having males and 50% chance of a person having female. So that's what you need to talk about when it comes to so sex determination, guys. These are the same genetic cross like these ones, ne? But now this one, the only difference which is there is that now instead of using, for example, they can say black, 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 and crossed with white, whereby black is dominant over white. So capital B, then small b, for example. But now here, if it is sex linked, it means that I will say the male is XY and then the female is XX, meaning that I have to carry these genes on top of this X. So meaning that whenever they say that it's sex linked, you have to use X and Y plus that allele they have identified for you. Please don't use X and Y if they don't say that it is sex linked. I repeat, don't use X and Y. In most cases, X and Y, these factors or these disorders, they come in hemophilia and color blindness. Hemophilia is the failure of the blood to clot. We saw it. So please don't mess it up. We go to the next one is I'll talk about it again. Blood group. When you come to blood group, we are doing a highlight, ne? Doing a highlight before we started the 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 the, the, the questions. Blood group. When you say blood group here we are talking about in blood group we are talking about um, the character which is controlled by multiple alleles. Remember, when it comes to blood group, make sure that when you say alleles, there are three: allele for A, allele for B, allele for O. Don't modify them. Don't write them like this. Don't write them like this. Don't modify these I's. They are supposed to be like that. There are three: allele for A, allele for B, allele for O. So, you will see that A has this homozygous or heterozygous. B has homozygous or heterozygous. AB is just like that, is codominance, and then O is homozygous recessive. Yes. So we need to know that. How many alleles? They can even ask you a question that can a person of blood group A marry with another person of blood group A? Can they produce a kid of blood group O? Yes, because blood group A can be heterozygous. The father is like this and the mother like this, so that this I and this I, they combine and then they produce O, blood group O. So it is possible. They can even ask you a question: why is blood group not a conclusive to determine or to identify the paternity or to identify someone's father or mother? Yes, it's because uh it's because I was just reading a comment. This one. Okay, it's because blood group is common among individuals. You understand? So blood group can only be used to eliminate some people that this is not a father, but it cannot be a conclusive way to determine that this is your father, this is your mother. No, it cannot be conclusive. This is what I mean. Let me show you what I mean. If they say that a person of blood group A married a person of blood group AB, ne? And then a child of blood group O was produced. Do you think this is the father? If the father, do you think these are the parents of this child? Definitely no, because blood group A, yes, blood group A can be heterozygous, but blood group AB, there is no way how this will bring blood group O. You understand? Remember, blood group O has two eyes, but now this one, it's only one eye here. Therefore, there is no way how this could be the parent of this kid. Maybe one parent and then another one. Yes. So we can only use blood group to eliminate some people. So we are gone. These are questions for practice. Dihybrid, when we're setting dihybrid, we can just bring a box like this and then we eliminate some of this and we ask you to fill in. Or we ask you to formulate the genotype of the parents, meaning that identify the parents' genotype. So in this case, you just have to use like this, capital B. Is there any other capital B? Yes. Is there small b? So it means that you don't repeat capital B. So small b. So meaning that if it's capital B, I have another capital B, so I can't. I have small b, yes. I have another small b, I don't repeat it. Capital L, yes. And then do I have another capital L? Yes, I cancel it out. So I go to small l, yes. Do I have any other small l? Yes. So it means that the parent was like that. Or we can give you this and ask you to form the gametes, meaning that I have capital B, capital L, small l, meaning that this is going to go with this. So I'm going to say capital B, capital L. Now this with this is going to be capital B, small l. Now this with this, small b, this with this, I said this with this, small b, capital L. Now this with this, small b, small l. You see that I'm able to produce the gametes which they produced from that individual. So that's what we usually ask you when it comes to the dihybrid cross. We don't ask you to go through all this, no. So we shall see the questions. Then when it comes to the genetic lineage, here it depends. This is where the problem is. This one, it depends whether it's caused by a dominant allele or is caused by recessive allele. You need to know whether it's caused by autosomal or is caused by sex-linked. If it is sex-linked, you need to know, use X and Y. If it is non-sex linked, don't use X and Y completely. However much they have talked about Joshua and Ronell and it's not sex linked, don't talk about X and Y. However much you know that Joshua is a male, and however much you know that Ronell is a lady, however much the box, you know that the box means a male, a circle means a lady, don't use X and Y because they didn't say that this is a sexy linked. For example, in this question, they didn't talk about that this is sex linked. They didn't talk about it. You understand? But it shows that it is caused by a dominant allele because the A is a capital letter. So it's caused by, so this condition is non-sex linked caused by a dominant allele. It can be non-sex linked caused by a recessive allele. I put a full video about this. If just in case you don't get it right, you can search it on genetics, ne? Then you end with Thunder Educ, then you'll be able to get. Someone's asking, how do I get a distinction material book? You just click on that link which shows that join, then you'll be able to join, and then you will get it. Yes, but they'll charge you something like 19 rand, ne? Yes, it is an online version. Or you request it from our offices, then we courier it. But now it is too late for you to do that. So it's better you use the one which is online. All right, we got the next question. So if, okay, Another question here, they can also ask you, how many generations are here? It's 1, it's 2, it's 3. How many kids were produced by Teboho, is Teboho and James. Uh, this, yes, meaning that if you look at this, how many kids, you look at this generation, but now they produce these kids, meaning that John just married Johanna. You understand? So this one is out of the what, of the kids which were produced by, by James and Teboho. Yes. They can ask you, what is the phylogenetic tree? We can ask you. Yes, just a tree which helps us to trace the disorders among the families. Yes. So, lastly on this, we see that these sex-linked disorders are very common in males than females. They are very common in males than females. However, it is they can ask you, why are males have this sex-linked disorder than females? So we know that males have have one X. If a recessive allele appears on one, yes, the moment the recessive allele appears, for example, is a small c here, the individual is already sick. While females have 2X, ne? If a recessive allele appear on one X, it can be masked by a dominant allele on another X. The individual is still what? Normal. Meaning that males need one small recessive allele for them to be sick, while females will need two recessive alleles. Two recessive what? Two recessive alleles for them to be what? To be sick. You understand? Yes. So meaning that females in this sex-linked disorder, females can be what? Females are carriers. They carry the disease. They don't show it up, but they still have it. You understand? Yes. So that is genetics. Then the last part of genetics which you need to know about is genetic engineering, so that you're able to go to our questions. Genetic engineering. Genetic engineering, basically manipulating the genes. You engineer means that you're a manipulator. You manipulate. If you manipulate, it means that we call it genetic engineering. Yes. So there are some advantages and disadvantages. These are mutations. We saw them. Yes, there's some advantages. Is expensive, interferes with nature. So these are just, you just need to memorize it and cram it, and yeah. Then disadvantages is expensive. Or advantages, we can use to produce medication like insulin. Yes, we can use it to control pests. We can use it to increase the crop yield. Yes. Stem cells. Stem cells, these are cells which have ability to divide and modify into other body tissues, meaning that they are not yet specialized. Yes. When you are still a ball of cell, you will not be able to see your head. Where's your head? Where are your legs? They are not yet specialized. Those are cells are not yet specialized. Yes, the sperm just fused the ovum, and then you mitosis occurs, and then you form a ball of a cell. There is no head, there is no what, it's just a ball. Yes, so those are specialized, the unspecialized cell, until when they become specialized. The same way, like you, I can ask you, who are you? You are just a student. But as you study, you're gonna start specialize in a specific area. That's why you are busy studying. We'll ask you, what do you want to be? I want to be an engineer, meaning that now you're gonna specialize in being in engineering. You understand? So at this time, you are just a ball of cells with nothing, no direction, until you get the direction of specialization. That's what it means. Those are the stem cells. All right, we continue. Cloning. So these stem cells, they have advantages. For example, they are very important in replacing the damaged cells. Yes, we can also use them for research. Yes, for research. Yes. So there is cloning, whereby cloning, you guys, you have to talk about cloning of a doll ship. Here we're gonna talk about which method is we use, why do we remove, why do we keep the nucleus, why did we throw the nucleus, how do you call maybe this ship, which this resultant ship is gonna, which ship is gonna resemble, yes, and then give a reason for your answer. So question like that. Uh, uh, I think many people are confused that they are writing tomorrow. You are writing on Tuesday. You will go to the examination room and then you will find nobody except you. Guys, you are writing on Tuesday, not tomorrow. Tomorrow, tomorrow is a religious day. That's what they said on the timetable. Yeah. All right, we continue, guys. So that is it. This one they are profiling just to determine the paternity test. Remember DNA profiling in DNA. Yes. So that's what we are doing. Yes. And then blood grouping, I also talked about that is can only help us to eliminate some people. So it is not conclusive. It cannot be conclusive to show that this is the father, this is the mother. So now let's go to the questions. Mitochondrial DNA is because now we only find it in the female. So the males don't have, remember the sperm, when it fuses with the ovum, it's only the head which fuses, meaning that the mitochondria of the sperm does not go into the kid. You understand? So that it means that we only have the mitochondria from the our female ancestors or female parents. We don't have those ones from the what? From the father. So if any mutation occurs, we can use it to trace it, and we know, oh, that, oh, we originated from there. Actually, we will see it in out of Africa hypothesis. So that is it. Now let's go to the questions and then we start answering some few questions. Yes, and then we will go to evolution. At this time, we were supposed to be starting evolution. All right, we are saying that uh, colorblind is a sex-linked disorder. You see, they mentioned it. It's a sex-linked order caused by a recessive allele. So remember we said that it can be caused by a dominant allele or by a recessive allele. When I say D, I mean a capital letter. It can be capital K, it can be capital D, it can be capital C, it can be capital what, whatever. I don't care. But as long as it's a capital letter, then it means that it's a dominant allele. Then it can be caused by a recessive allele, small d, meaning that you have to be with the two of them to be sick. Here, dominant allele, you just need to have one to be sick. But if you are recessive, you have to be with the two for them to be what? Remember, the moment there is capital D and a small d, the capital D will mask the presence of the small d. Then it means that you're going to be a carrier, not what? Yes. It's going to be a carrier. You won't be sick. You'll just be a carrier. Yeah, that's what it means. So now let's start. Uh-huh. Um, okay. Give give the phenotype of senzo. If you look at here, senzo. Senzo is here. But now I need to know, they say that give the phenotype. The phenotype, remember guys that a circle means a female. A box means a male. Circle with the shaded means you are affected with that disease. In this case, you are colorblind, female. A circle with a box with this, you are colorblind, male. So now, since I know that, I will come back and then, since they gave us the key, it is simple. Senzo, they say the phenotype, physical appearance, meaning that Senzo is a colorblind male. Done. Tuli, genotype. You want the genotype of Tuli. But now, I want to know who is Tuli until I go to the kids. So I will say that Tuli is female. You understand? So Tuli can be capital D, small d, or capital D, capital B. because they are saying that Tuli is normal. You see? And then this one is going to be capital D, small d, because it is sick. So Y doesn't carry. Don't forget that Y doesn't carry anything. Y doesn't carry anything. I repeat, Y doesn't carry anything. So since I know that, then now I'm going to say that this with this, so it's going to be this. How did I produce Senzo? How did I produce Linda? So Linda is female, meaning that it's like this because it's caused by recessive allele. So I will need 2 of them. And then Senzo is also sick. Ne? You see? Meaning that, remember, Y doesn't carry, so I will only need 1D. Please don't put anything on Y. Yes. So now I will say that if this came from the father, where is the second one came from? Came from the mother. You see? Meaning that the mother was like that. If this one, obviously, why come from the mother? It can't come from the father. Sorry, come from the father. There is no way how it's going to come from the mother. Then the remaining one also, it will show that it came from the mother. Therefore, Tuli was like this, not like this. So that's how we identify the genotypes. That's how we identify the genotypes. Don't just rush. It can be, unless there is no option for the babies. Ne? You understand? Like, for example, these people, there is no way how you're going to know that it's like this. If, no, the male we can know. Yes, the male, still the male, because the male is sick. Yes, so the male we can know. Because the moment is normal, there we go. There is no any other option. So this one can be this, yes. But because the male is sick, then we will know that, since the male is sick, then definitely this one is going to be like this. There is no way how this one is going to be like this. Then if this came from the mother, where is the second capital D is going to come from? Yet the male is already sick. We know that the male is like that. So here, you don't rush. It's just application. Guys, this question is always application. So you just have to take your time. Be slow. There is no need for you to rush out of the exam when you have not finished. You are rushing to do what? Guys, I repeat, and I said it even in paper 1. There is no need for you to leave a question which you can do and you leave it unanswered. For what reason? Because you are rushing. Okay. Never. Don't do that. You are rushing. Let me continue, guys. Please don't rush. They're not chasing you. The examiners said 2.5 hours. They are not fools. No, they are not. What do I mean? They are not fools to tell you that use 2.5 hours. They are not. Then why are you rushing? 1 mark per 1 minute. That is how the paper is set. 1 mark, 1 minute. But there are some marks, that 1 mark, which is going to be very difficult for you to use 1 minute to finish it, to get it. So there are some questions which are easy to get. So use the other one, collect more time to finish up these questions which will require you more time to get that 1 mark. That's how the balance is. So please, guys, don't rush. Explain why are generally more males, we explain this, more males than females with color blindness in this population. Because males have one X chromosome. When the recessive allele appear on the X chromosome, the individual is what? Is sick. Meaning that males only require one recessive allele for them to be sick. While females have 2X, if a recessive allele appear on one X, it can be masked by a dominant allele on the other X. The individual is still fine, meaning that he's still normal. In other words, females require two recessive alleles for them to be sick. But sick of what? You come back. For them to be colorblind. You understand? So it means that you have to explain based on the question which they have given you. Don't rush. Then they are saying that Molly married a normal male. Married a normal male. All right. Married a normal male. Molly. We need to know who is Molly. Molly. Where are you? Okay. Molly married this Molly. Okay. Now we don't know who is Molly. But since from the mother, we know that the mother Tuli is capital D, small d. And then this one is small d, y. Remember, y doesn't carry. Definitely Molly can't be homozygous. So it means that Molly was capital D from the mother and then small d from the father. That's why this person, meaning that this person is normal but sick. Normal but a carrier. They said married a normal male. Normal male from where? I don't know. From the street, from the hospital, from the clinic, from the what? I don't know. Capital D, small y, and y, meaning that it's a normal. Now look at this. This can be P1, F1 because they didn't say that they came from up, or because Molly is on the second generation. It might be, you can use T2, F2. Yes, I hope you get it. So meaning that I'm gonna say, remember our first step, ne? T1, F1. But we say that because this is a second, Molly is on the second, so we can say P2, F2. You see, you get 1 mark. So you tell us parent 2, second inferior generation, phenotype. You see, this is how we mark. Yes, phenotype, you tell me. Phenotype of Molly, we say that Molly is normal. Normal female. Normal female. Married who? Check. I told you, you get this thing from the question, normal male. So, married a normal male. So you get 1 mark here. We give you a mark. You get another mark there. You see this mahala. Then you go to genotype. I don't know the genotype. Meiosis. I don't know the meiosis. Gametes. I don't know the gametes. Fertilization. I don't know the fertilization. You leave. You go to another question. Still, we shall come and do like this. We give you a mark. You have 3 marks now. Okay. If I know the genotype, now meaning that I have 3 out of 6. So if I know the genotype, what did they say? They said Molly. And then we saw it here that Molly is capital, is a carrier. And the other guy is normal. Therefore, I will come here and say Molly, who is capital D. Molly, who is a small d. Crossed with normal male. A male is like that. So normal. Remember, Y doesn't carry anything. Then meiosis occurs, capital D, small d. And then here, capital D, Y. Remember, these are gametes. I always tell you that don't leave them to be like at the beach. Dress them up because people at the beach are always naked. All right. So don't leave them naked. Dress them up. Then, from there, you can use a cross. Now, this time, let me use a Punnett square method. Draw a box. You draw a box, bring this here, and bring this here. So it means that I'm going to say capital D, Draw a line, and then you bring this here, small d. You see x small d, correct. You bring now this one here, capital D, draw a line, then you bring this one here, y. You can circle them if you want, even here. And then we say this is called fertilization. So we already got that mark. So you remember now you have that mark, you have that mark, meaning that now you have 5 out of 6. Then now you say this and this, so capital D, this and this, capital D, small y, this and this, capital D, small d, this and that, capital D, small y. You see? So please don't forget that. So we call this one offsprings. Yes. This message goes to the people who are able to do this. Thank you so much, guys. And the people who have been with us from the beginning of our journey. Meaning that I mean those people who are under our private classes. And those people who are inviting us for special classes. Thank you so much. We love you so much. All right. Thank you. We continue. So from there, what is the question saying? You see, once they bring this genetic cross, guys, just know that those are free marks. Mahala. Use the genetic cross to show the possible genotypes and phenotypes of their children. Meaning that I have the genotypes now. So I just need to know what is what. This one is a normal. This is now a phenotype. Normal, female. How many are they? It's 1. Yeah, normal female. They say the phenotype. Two normal female. This one is normal. This one is normal, but a carrier. But normal is normal. I don't want to know whether it's a carrier or what. It's normal. And then this one, normal. 1 normal male. And then 1 colorblind male. Don't say 1 sick. Sick of what? People don't know when they are writing. Just read your timetable properly. People don't know that they are writing on Tuesday. People, you're making me happy, ne? Hey, by the way, guys, did you know that the reason why teachers don't get old is because of you guys? You give us stress, you give us happiness, you make us smile, you make us become angry. Like, we experience every it's not always like we are angry. You can be angry. Maybe, for example, I'm just giving an example. For example, you leave home, you are angry. I'm just giving an example. You leave home, you are angry. You reach school, you are busy teaching, and then someone give a statement. Oh, you are busy marking. You see someone give a answer, you laugh. You see? Thank you so much for making us, yeah. And you can also say same story. You can make us angry. You leave school, you are angry. You reach home, and then they also make you happy. So it's 2 ways. So thank you so much, guys. Uh-huh. You students, you make us happy. So, guys, that's the question. Let me look for you another question of genetics. Another question of genetics, and then we smash it. Should I give you this one? Even this one. Let me give you this one. Yes. Okay. This one. Let me check and see. Yeah. Let me see this one, guys. This one is page 29. Page 29. Let me see this one. The type of difference in humans occurred on a single allele. Diagram shows the difference. Okay. 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 Genetic cross to show 60. Okay. Okay, there's that one. I think I'm gonna give you also this one. So that we are able to go to You're able to go to. Hey, there's a question which I didn't answer, ne? And I wanted to answer it. I forgot. I don't know how did I forget it. But okay, I'll do it, ne? I'll do it for you guys. I was forgetting it, ne? I was forgetting it. Okay, it's fine. I'll sort it out. I'll sort it out for you. Okay, let's answer that. They're saying that I should give you the one for color blood. I mean, for dominance. Okay, it's fine. The one type of deafness in human is carried on a single allele. The diagram below shows the inheritance of deafness in a family. You have Liz, Paul, what? Okay. How many generations are here? It's 1, it's 2, it's 3. So 3 generations. Ne? Yeah, cool. So, then they are saying that how many generations are represented in the pedigree? Children, how many children of Paul and Liz? Children of Paul and Liz, it is 1, it's 2, it's 3. This one Anne just married to Bob. So there are 3. They are saying that which phenotype is dominant? Which phenotype? Now here they didn't tell you whether it's dominant or recessive. Okay. All right. Sibusiso, you are starting to smash the things. Smashing the paper. Okay. Smash. All right. We are saying that which phenotype is dominant. So we need to find out which phenotype is dominant. If you have this, Paul, remember this is not sex linked. If you have Paul and Liz, yes, then you produce someone who is this. You won't get it. Let's look at this one. If you have Bob and Anne, and then you are saying that Bob is able to hear and Anne is able to hear, but they produce people who are deaf, meaning that, you see, this one is deaf, meaning that, because if it was a recessive, which letter did I use? Let me just say deaf. Let me just use D, meaning that small d, if this was dominant, ne, there is no way how this is going to come from this without a D. So it means that, let me clear it up. It means that Bob was capital D, small d. And also Anne was capital D, small d. So that they produced small d, and then this child was produced. You understand? Yes. Because if this was the recessive one, for example, small d, and this one is small d, ne? There is no way how they're going to produce a capital D here. So meaning that the deafness was recessive. The deafness was recessive. Use the offspring Bob and Aibo. It's the question. It's the same question they have explained. Yes, because here you can't explain using this, because already this one there is one which is shaded, and also here there is one which is shaded, so you couldn't. The only option was this. That's why I also use this, meaning that this individual, this male individual, remember it's not sex linked, so I can't use X and Y. You understand? Yes, because they didn't say that sex linked. I told you that if sex linked, they will tell you that sex linked. So it means that this, they said which letter do we use? Yes, it means that when Bob and Anne married each other, they produced an individual who is deaf, yes, which is recessive, yes, meaning that this deaf person received one recessive allele from the mother Anne, and one recessive allele from the father Bob. Yes. So meaning that the two, they were heterozygous. Bob and Anna and Anne were the heterozygous. They passed one recessive allele from each to the male kid. Therefore, the male kid uh, uh, became deaf. Use letter A to represent the dominant allele and letter A to represent the recessive allele and give all the possible genotypes for hearing. Give the letter A to represent the dominant allele and the small letter A to represent recessive allele. Give all the possible genotypes for hearing. Remember now hearing, we said that hearing is dominant. So they're saying give all the possible alleles. Oh, no. Give all the possible genotypes. Meaning that you can be heterozygous. Yes. So you can be homozygous, dominant, or you can be heterozygous. So this person is able to hear and this person is able to hear. So you'll get a mark here and you get a mark there. So you are able to get it right. Use a genetic clause to show the gender, how gender in human offspring is determined by sex chromosome. So it means that here you just explain gender determination. This. Yes, you just explain. I just explain. Yeah, this. Just explain. Just write the same way. If because they say the genetic cross, you just say P1 F1. Now you just say P1 F1. You say phenotype. You say male crossed with female. Then genotype. You say X and Y. Then you say XX. Then you say meiosis. You cross. You say gametes, you form the gametes. You say fertilization, you cross, you get the fertilization. You get the offsprings. So these are the offsprings. So basically that's what they are looking for. They don't want you to explain using this formatting. They say that use a genetic cross. They don't want you explanation. They just want a genetic cross. So that is it. Please guys go to the paper where you know how to do a pie chart, ne? Yes. Please go to the paper when you know how to do a pie chart. Percentage increase and percentage decrease, I talked about it. So I'm not going to repeat that one. Yeah, let me just do this small question. This small question. Yes. This question, page 37. Okay. They are saying that diagram below represent DNA profile of a child, of children and their parent. Only 2 children are biological. Okay. Identify 2 biological children. So when you talk about biological, you look at, remember, when I take off the one for the mother, the remaining bars must correspond with the father. If don't correspond, then it's not. So I will say that if you look at this, is this goes with that, nothing. This goes with that. Is it right there? I don't see it right. You use a ruler, then this will go with that. This, I hope it goes with that. Then this goes with that and also goes with that. Then this goes with that. This goes with that. Meaning that you see that, oh, this one also goes with that. Meaning that you see that all of them have been covered, and also all of them have been covered between the father and mother. This one, there is some which remained. So it means that is this one and this one who were the biological children. Yes. Stage 3, other uses. You see now, you don't talk about paternity test. You don't talk about paternity and maternity test. Why? Because they have talked about this. So it means that you can talk about that forensics. You can talk about identification of genetic disorders. So that's what you need to talk about.
Hello, sir. You missed the other one for the father and the prior.
The father, no, it's not corresponding. It shows that it's not. It's you see that the line is not straight. If it was going to straight,
Okay.
you see.
Mm-hmm.
Yeah.
All
So
right.
it means that Priya is an adopted, is an adopted child. Then we go. Thank you. Then we go to this question. Brown enamel of the teeth is brown enamel. So it's you having brown teeth is not. It's not because we are eating a specific food. It's because of genetics. Yeah. Right. Right. They're saying that. Sir, you're not gonna do evolution. Eh, I'm going to do evolution now. This is the last question. I'm going to do evolution now. Yes. So you have yes. Brown enamel. Remember, evolution is not that complex. After we go through it. Yes. Okay. Brown enamel of the teeth is a sex trait. You see, they mentioned it is sex trait. A dominant allele on X, you see, they mentioned it that it is caused by a dominant allele. You see? A dominant, I don't know whether it's A, okay, or C. Cause brown teeth, okay. A dominant allele on brown, so it means that it's just explain why more males than females have white teeth, because now it is caused by a dominant allele, meaning that the moment you have a small d or a small a, yes, you males will require two recessive alleles. So I mean one recessive allele for them to have white teeth, and then females will require two recessive alleles for them to have white teeth, because this is caused by a dominant allele. So meaning that it is very common in males than females. For them to be having white teeth, yes, the males will require one recessive allele which is going to go on X. The same thing which we talked about color blindness is the same thing which they're talking about here. And then females, for them to have two recessive, you need two recessive alleles. For example, is A for them to have white teeth, meaning that in this case females, you'll find out that females have more brown teeth than the white teeth, because white teeth is caused by a recessive allele. So it's very common in males. So they're saying that a man who is brown teeth married a lady who is white teeth. You see, man is brown teeth, meaning that if a man is brown teeth, it's gonna be okay. They're saying use A for brown teeth and small b for white teeth, meaning that the male is gonna be, they said brown teeth is gonna be like that, and then the female is gonna be capital T. They said white teeth, meaning that small b and then small b. You see that females, for them to be white teeth, they require two recessive allele for them to be white teeth, because they said white teeth, and remember white teeth is recessive. So now what are they saying? Use a genetic cross to show the post. So you see that our 3 marks are there because they said this crossed with this. So I'm gonna say brown, guys, and then you fail these things. There is no way you're gonna fail these things. Make sure that you get mark. You get me marks. Yeah, those marks are not yours, they are mine. Make sure that you get me marks. Make me happy. All right. So we are saying that P1, F1, it's the same thing. Phenotype. So you get that 1 mark. You say phenotype. Phenotype. The phenotype, we said it. It is brown teeth, white teeth. Female, male, brown teeth, crossed brown teeth, crossed with male, female, white teeth. Female, white teeth. So you get another mark. Then you say that genotype, meiosis, gametes, fertilization, then you get another mark for fertilization and meiosis. So you have 3 out of 6. Is it 6? Let me check. It's always 6. 6. So now after that, you have already identified them here. That the male is capital B, Y, and then the female is small b, and then after that meiosis occurs, and then after meiosis occurring, I'm gonna have capital B, and then I'm gonna have Y, and then here I'm gonna have small b, and then small b, and then after that I can use the Punnett square where fertilization is taking place, or I can just cross. So depending on, make sure that if you cross properly. Then is that capital B, small b, then is that small b, y, and then is that small b, y, and then we call these offsprings. What is the question saying? They're saying that use the genetic codes to show the possible genotypic ratio, the phenotypic ratio. They want the phenotypic ratio. So the phenotypic ratio, you have two female. They want phenotypic ratio. So the phenotype 2 female, brown teeth, and then 2, it's gonna be 2. How many? 2 males, white teeth. So since they want the ratio, is gonna be 2 to 2. But if you leave the answer like this, we're gonna cross it, because the ratio is supposed to be simplest form. So divide by 2, so it's gonna be 1 to 1. So the answer becomes 1 to 1. The ratio is supposed to be simplest. If you don't do that, 0. Let's go to evolution. Let me see if I've answered what I wanted everywhere. Yeah, there's another question there is for, okay, it's the same thing. Okay, yeah, can't exhaust 33. Okay, on the wrong page. 33. Cool. Yes, it's just 1 or 2 questions. So they're saying that how many generations are shown in this diagram? It's 1, it's 2, it's 3. So 3 generations. Give letters of unaffected male. When males are like this, meaning that they're saying give letters of unaffected, letters or letter of unaffected male. Males are boxes, so I have only H. H. Saying give the genotype of A. A is a box affected male with what? Moya. So it is male.
Someone asked the question.
I see them. I see the questions. Don't worry, I see the question. They're saying that moya, there is a male with moya. So A is moya. A is moya. Yes, moya is a disorder. Okay, wait. Moya is a disorder caused by a dominant allele, R. You see, this is caused by a dominant allele. R. So since they say that the one who is shaded is an affected male, so it means that this person A is a male with moya moya. Remember, it's a male with moya moya. But they wanted the genotype. So since they want the genotype, remember they didn't say here that this is sex linked. So I don't expect you to say X and Y. That's what I said. I don't expect you to have X and Y. So since it is caused by a dominant allele, listen carefully, is caused by a dominant allele. So it means that this person can either be capital R, small r, or capital R, capital R. Let me, this is what I wanted. Remember, we said that it can be caused by dominant allele or it can be caused by a recessive allele. But here they didn't say that this is X-linked, so don't use X and Y. Is it clear? So now, if it is not caused by a dominant, it's not caused by a recessive allele, so it is caused by a dominant allele. They said it here. So it means that this person is capital R, or capital R, small r. But I need to find out who is that person. This one, meaning that is a small r, is normal. So the person who is normal, meaning that here is a recessive one. Because it is caused by a dominant allele. The one, all the questions we have been doing, they were caused by a recessive allele. But this one is caused by a dominant allele. That's why I say that you need to know whether it's sex-linked, dominant allele, sex-linked, recessive allele. Non-sex linked, dominant allele, non-sex linked, recessive allele. Yes. So one will come. So now, since now I know, remember I can't confirm this until I go to the kids. I have 2 kids. Remember, this just got married and this just got married. But now what is the genotype for this? So this person can be, yes, this person, I need to go to the babies and find out, can be capital R, small r, or capital R, capital R. Is a what? So I don't know. But this one is recessive R, because it is normal. So if it's one normal, then it means that remember it's caused by a dominant allele, meaning that you have 2 recessive alleles. So this person E cannot be like this because one allele came from here and then one allele came from here. So since this person is like this, one came from here, and then where is the second one coming from? Came from the father. So the father can't be like this because of this. So the father is like this, so that this second recessive allele came from the father, meaning that A is capital R, small r. A is capital R, small r. You see, now like this question might take you, if you don't know, you have to get it from there. You must always look at the babies before you conclude this. Yes, so it can take you more than a minute, so that now the other time, which is you obtained from the faster question, maybe like a terminology, you are able to get them. They say that individual letters of individuals not biologically related to A and B. Who are those letters of the individuals who are not biologically related to A and B? We see that this one got married to this, and then this one got married to this. So meaning that A and B only gave rise to these 2 kids. So the answer is C and F, because those ones, they just came to get married. So it is C. We said it's C and F. Evolution. Ah, yes. Evolution. Evolution does not take too much. It's just applications, application questions. So you guys, you need to know the application question. Let me just look for you nice questions here. Mm. So I'm gonna say which page. Yes, there's nothing evolution there. So there is evolution in there. Okay, this one is a wrong page. Okay, let me check and see which one is evolution. Yeah, take some water just for a minute. A minute, just water. Stretch 1 minute. 1 minute. Yes, take some water and then yeah. Hmm. Okay, guys. Okay, guys. Let's go through the evolution. Evolution. Let me just do a small highlight. 1, 2, 3. Then after that, we are able to answer. Oh, Winnie. Oh, thank you so much, Winnie. I'm just seeing your message now. I wrote paper 1. It was so nice. No, you are making me now happy. You are making me happy. All right. Let's start. Guys, don't forget that in evolution, we have what called human evolution and evolution by natural selection. So let's just use. It's getting too late for me. Let's just use. Let me highlight for you, and then after that we do some few questions which are going to guide you to get the marks. We go. 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 Evolution. So when you talk about evolution, is basically increase in the complexity of an organism. So you need to know that when you talk about evolution, is talking about increase in the complexity of an organism. So that is what called evolution. So in this evolution by natural selection, best of it, there is nothing, completely nothing we're gonna bring and ask you to label. Nothing. It's 100% application. That is evolution by natural selection. And don't forget that evolution by natural selection, it has a lot of marks. It has a what? A lot of marks. Uh-huh. So now let's start. By the way, this discussion could also it helped those who are writing May, June, but not finals. But now, starting from this topic, you are not going to be asked about evolution. You only be asked about evolution in the next what prelims. For those people who are writing from Gauteng, those people who are writing from other provinces, yes, some of you start with evolution. So cool. Let's start. Evolution, we need to know basically what you need to know. What is a theory? What is a hypothesis? That's it. A theory, hypothesis, a tentative statement that is subjected to be. Who is that now? You are making noise. Yes. So we are saying that we need to know what you call a hypothesis or a theory. Basically, a tentative explanation or a phenomena which is subject to be verified, or we will need to investigate it about. Yes, that is a hypothesis. They can bring it in a terminology. Then a theory is an explanation of a phenomena which has been proven to be correct, meaning that is supported by the hypothesis, tested hypothesis, experiments, facts, all that. Yes. Then fossil, we will see it in human evolution. Here we brought this, that distribution of species in the different parts of the world. This is what they were asking you by geography, and then I gave you the answer. Then comparative anatomy, this one we call it modification by descent. We like to ask it in terms of terminology, modification by descent, which is called comparative anatomy. We call them, these structures are called homologous structures. Yes, so we compare the 5 digits. That's why they call them pentadactyl limbs. Penta, which means 5, dactyl, which means digits. So that is it. Then we go to the next question. We go to the next question. Causes of variation. We said variations. Okay, before we go to the variation, a species is a group of organisms that is able to interbreed to produce a fertile offsprings. They must have similar characteristics. Yes. Then you have a population is a group of organisms of the same species. They must be of the same species. Yes. So meaning that if they're not of the same species, they're not a population. Okay, we are done. Causes of what is variation is the difference among organisms of the same species. That's what it means. And then when you look at causes of variation, that is crossing over, random arrangement of chromosomes during metaphase, random fertilization, and random mating. Yes, that is also from meiosis. They can even ask you that what are the causes of meiosis? What are causes of variation based on meiosis? So you must be able to answer that question. Meaning that based on meiosis, then you talk about random arrangement of chromosome during metaphase, and then you can talk about crossing over. Yes. So variation can be grouped into two, that is continuous variation and discontinuous variation. Continuous variation, it has an intermediate. You will see that it's continuous. It's called continuous variation. You have very few people who are very short, very few people who are very tall. Most of them are medium, meaning that the intermediate character has many people. Very few people who are too dark, very few people who are too light. Most of them are just in between there. So it's continuous. We have the dark one, and then we have the very light one. We have the intermediate, meaning that it's continuous. So basically, some of the examples are things which we are born with, and they are what? They keep on changing. The size of your shoe, the way you are born up to now is different. Your body size is different. So that is continuous. It keeps on changing. So it's basically caused by environmental factors. Yes. Then discontinuous is basically caused by the genetics, meaning that you are born with it. For example, there is no intermediate. If you are color blood group A, your blood group A, you will never change to blood group AB. Even if they take all the blood from your body, they put blood group AB, still will be blood group A. So we don't have any intermediate there. So it is discontinuous. Basically, this character is caused by genes. Then those are the simple questions which can come in objectives. So we have two theories which we need to look at. That is Lamarck's theory, Darwin's theory. Lamarck's theory has two laws, the law of use and disuse of the structure. He says that the law of use of a structure, the more you use the structure, if you use the structure frequently, it becomes bigger or well developed. And then if you don't use it, it becomes smaller and finally disappears. Whenever you are explaining how did this came up with this, you have to use those two laws. The second law is the inheritance of the acquired characteristic. You understand? Meaning that the characters you get from the lifetime are passed to the next generation. If you're a bodybuilder, you're going to produce a kid who is a bodybuilder. You understand? Yes. So you use an example of giraffe, but the other examples we can use. So it means that here you have to apply. Don't use exact the same. If they bring the snakes, use the snakes. If they bring what? Use that. Make sure that you use based on the what? On the question. Then we have why many people, even some of you are busy saying, why is Lamarck theory not accepted for many scientists? Because he says that if you want to be longer, you have to stretch your neck. You keep stretching until you become longer. So not saying that characteristics are not obtained during lifetime. They are not passed on because they are supposed to be passed on. No, characters are passed on because of the what? Of the genetics you have. So he may say that no, organisms evolve because they want. It's not like that. Organisms don't evolve because they want. Organisms evolve because of what is happening in the environment. You understand? Because of its genes. So here I say that the giraffes, all of them had short necks, meaning that there was no variation. Then they started to stretch their necks due to the scarcity of food. And then they stretch their necks so that they're able to obtain these leaves from the tall tree plants. Then because they are using the necks became longer. You see? They're becoming longer. And then now they pass this character of the long neck to the next generation. So the offsprings were born with the long necks. So it means that if you want the long neck, you have to use it. So these are just examples of giraffe, but the question will not come specifically for giraffes. Explaining how snakes lost their legs. Meaning that the legs were making them not to use them. So they kept on not using the legs. So they're able to move faster. Now the legs became smaller and then they disappeared. Meaning that in this case, he used the law of disuse. Remember the law of use and disuse. You either use the structure or you don't use the structure. Okay. Then there is not enough evidence to show that acquired characteristics are inherited. Yes, there is no. Now, in this case, the question is going to come like this, and then you write it like this, exact like this. We're going to cross it. This is the answer. But if you write it like this, we're going to cross it. So in this case, you're going to say that there is no enough, for example, based on unless it's a generic question, they ask you, explain why Lamarck's theory is rejected. Then you can say there's not enough evidence to show that acquired characteristics are inherited. But now if they are talking about, for example, the giraffe, then you tell us that giraffes, there is not enough evidence to show that the long necks acquired, yes, were acquired during lifetime, were inherited. You see, that is the acquired characteristic. So it means that you have to be specific. Which character are they talking about? That's when you join them, please. Organisms don't evolve because they want. Organisms don't evolve because they want. Organisms evolve because of their nature. Do you think you are here on this YouTube at this time, on this Zoom at this time, supposed to be eating or in the bed because you want? No, you are here because of the nature. You want money because of the nature. You know that you don't study problems. There is a force which is forcing you to do that, not because you want. You think me, I'm here because I want too much. There is a force which is behind me that, I want my kids to pass. You see, the nature is forcing me that, you have to make your kids to what? To pass. Not because I feel like going there and then teach. There is a force behind. So organisms don't evolve because they want. The organisms evolve because of the nature, the natural selection, because of the environment they're in. The only option you have to modify to suit that. That's why the organisms evolve. Yes. All right. So you need now Lamarck. Okay. Then Lamarck, Darwin used the theory of natural selection. Yes, there's no that. For him, Lamarck said that there is no variation among organisms. Darwin said that no, there is variation. There are those people who are okay. Those giraffes which were short neck and those which are long neck. Those which were short neck, they died because they couldn't reach the tall tree plants. While those which are long necks, they are able to survive because they were able to get the food from the tall tree plants. And then this, because they are being favored, this character is called natural selection. You understand? So these giraffes which had long necks, the character, the gene for long necks, they pass their gene to the next generation. So after many generations, those which all giraffes had the what? The long necks. So Darwin at the beginning says that there is variation among the organisms. Lamarck says that no, all of them they are the same. If you want to be different from others, you do what you want to do, and then you become different. That's what Darwin says. I mean, Lamarck said. So, um, how did Lamarck explain the giraffe? He said that there is genetic variation. You see that giraffes, yes, had long necks. As a result of the leaves. So don't forget that the question won't come specifically with the giraffes. Being available only on higher tree plants, okay, the giraffes competed for these leaves. And then the shorter necks, those ones which couldn't reach the food, they died. While those ones which had long necks, they survived. And then this is called what? Natural selection. The allele for long necks was passed to the next what? Generation. And eventually all of them, they had long necks. Simple. So these questions are application questions. You have to be keen when you're answering these questions. Punctuated equilibrium and gradualism, I explained it. It said that in punctuated equilibrium, there is long period of no change and short period of rapid what? Change. Meaning that you don't need an intermediate. For this mouse to change from this line to this line, it just requires a short period of time. And then it stays long without what? Changing anything. While this one, gradualism, it keeps on changing every day, every day, every day. At the end of the day, you see that, there is a change. So it requires a lot of time here. While this one has short time. Here there is intermediates. We have the intermediates here. Here there is no intermediates. So we usually ask you that question. Artificial selection, basically you select those which are able to survive the desires you want. Then it's being passed. You allow them to mate. You allow them to have sex so that you're able to produce another kids. That is artificial selection. You allow them. Those which don't have, you deny. You deny because you see that that breed is not nice for you. So you make them to be left out. So that is artificial selection. So we're gonna see some questions. So they can ask you the difference between natural selection and artificial selection, differences between Lamarckism and the Darwinism. So basically in artificial selection, you will find out that human, they decide who. And then natural selection will decide, the nature will decide who is supposed to be passed to the next generation. So this thing is very common in wild, but nowadays it's not common in human beings. Why? Because of the technology. The technology is making the things known to. God these days don't die because of the nature. No, because there is medication which is going to make. It's not that the best suit is going to is going to survive. No, if just look. You think the best, the person who is most clever is the one who gets the job first? Who told you? These days is artificial selection. So that, oh, this one, I want this one. You see, hey, the moment the human is there, ah, they will look for something, even if you have all the qualification. They say that when I want the other one, I want the other character, not you. However much you have the same qualification. So it is the nature. It's not nature to select. It is what? It is a human. So yeah, guys, this question, make sure that you know it properly. So this is the first point I'm going to waste too much. Not waste. Talk about. Let me just use maybe 5 minutes to explain it. And yeah. And then after that, we shall go to, what do you call it? We shall go to present day evolution and then human evolution. Then we do a lot of questions and then yeah, we wind up. Okay, here, guys, when it comes to speciation, formation of new species, remember, in this case, please don't make a mistake to go when you don't know this, because these are marks for free which are going to come. So you have one population, yes? So you tell us that there was one population. The population is split into two. You tell us what made them to split. Maybe a river. You tell us. They will tell you in that question that it made them to split. So you have A and you have B. You understand? At first you had maybe A. You understand? Then now, because of this river, these organisms will not be able to cross to that side, and these organisms will not be able to cross. Therefore, I will say that there is no gene flow between A and B, between these two populations. And because of the different now areas, they're going to experience different environmental conditions. Meaning that natural selection is going to occur independently or differently. And that, because of that, is going to result in that these ones, they're going to modify differently from this. Meaning that they're going to become genotypically and then phenotypically different. You start with genotypically, and then phenotypically different. Meaning that now, this A will no longer be like A. It's going to be now A. And this B is also going to be like B. It's no longer the same way. So they become genotypically and phenotypically different. So even if you bring them, you mix them, A and B, you mix them together, they won't be able to interbreed, meaning that they have formed new species. So now, it can be two. Now, if you look at this, if this is A, you started with A, and then at the end of the day, you have A and B, meaning that now only B made a new species, you see? Meaning that A remained with the same species. What made the A to remain the same species? Meaning that the environmental condition which was here is the same as the environmental condition which was there. Or both of them can change if the environmental conditions are different. Then both of them, they can change, they become new species. Meaning that two have become new species. So a question can come as generic or a question can come as not generic. Meaning generic means it's general, meaning that it is you just write exactly the way it is here. But to know the application, you need to know this. A population of a particular species may be split. Oh, now this one, a population of a particular species split into two. You understand? Because now into two, depending on which, if they say three, if they say four, so depending on what they have asked you. You understand? Yes. So a population of split into two. And then you tell us what made them to split. By a river, by a mountain. And tell us geographical barrier. We don't want to know. Unless it is a genetic question, whereby they didn't give you an example. They just ask you, describe the process of speciation. Now you write exactly the same way, like this, nothing else. So a population of species split into two by a mountain. As a result of the two parts of population cannot interbreed, meaning that there is no gene flow. Gene flow means sex coming from this side to this side. Yes, there is no sex, there is no gene flow. So then because of their different, now natural selection will occur independently due to the different environmental conditions. Then they are saying that as a result, yes, two population became genotypically and phenotypically different. You start with the word genotypically, then you talk about phenotypically different. Even if the two populations are mixed, they cannot be interbred to produce a fertile offspring. Please don't use the word viable. We no longer are accepting it. Fertile offspring. One, now you tell us either it's one or both. In this case, it's both. They have become new species. Simple. So it's the same story here. You see that they are split. In this case, it's a box. Yes, and they are feeding on different foods, and then they made them to change. There's no gene flow in between. Natural selection occurs independently. Now, if you mix them together, they cannot interbreed to produce a fertile offspring. Then the two that become new species. So please don't go to the paper without knowing that. Then, so all these are the same. These are just mechanisms which prevent organisms from breeding. Yes, we call them reproductive isolation. You can be separated by courtship. The way how you date is different. If you look at the dog, it's different from the cat. It's different from except humans. You humans, they copy. You hear dog style, cat style. You people, but humans, hi, they're saying that breeding different seasons. You now you breeding the other time, the other one breeding the other time. So there is no way how they're gonna do the. I gave an example of someone yesterday, for example, when you are ready in you let me just leave it. You have adaptation different pollinating agents. You see there are some insect pollinated, wind pollinated, bird pollinated. It depends. So you see that these plants won't be able to interbreed. Then you can also have sex. You have the baby, but the baby is infertile. Still, it is preventing you from having babies. Like a dog, the horse and the donkey, when they cross, they produce a mule. A mule has 63. How is the 63 gonna divide during meiosis? You see why? Because the horse is 64, and then the donkey is 62. So this one, when it divides, it produces 32 during meiosis. This one produces 31. You understand? So when you combine, you produce 63. Now, how will the mule be able to produce? How is it going to divide when during meiosis? How is it going to divide? There is no way. It's going to produce 31 and then the other one 32. I don't know. So that's why it becomes infertile. So Galapagos is still the same thing, natural selection. This is the present day evolution. This is the present day evolution. How do insects, how do viruses, how do mosquitoes, how do bacteria become resistant to their either insecticide or RVs or antibiotics? This is present day evolution, meaning that's why it's called present day evolution. meaning that there was variation among these mosquitoes. Some which are resistant, some are non-resistant. So when you apply DDT, you see there's variation among these mosquitoes. When you apply the DDT for the first, it will kill most of them, but some of them will be resistant. So the resistant one will survive. When they will survive, they will reproduce in large numbers. So their population will increase. Continuous use of DDT. DDT is an insecticide. Will have little effect. It's the same thing which happens to people who are suffering from diseases which are linked to bacteria. Ne? That's why it is not easy to get antibiotics. They first tell you go what, yes. Because some people they misuse them. And also people who are suffering, who are having the HIV and they don't take medication. It's gonna be a problem. How? These viruses or these bacteria, they're gonna mutate in such a way that if you don't take them in the proper time or proper doses, when you see that you feel like you are fine, you stop. They're gonna mutate in such a way that when you take the same drug again, it won't be effective. Why? Because you did not take, they have mutated, they become the resistant one, they survived, and then now they have reproduced in large numbers. Even if now you take the medication, you will never be treated. That's why now like people who are they change them from, okay, they say that okay, now this drug is no longer working, then they give you a stronger one. So you're gonna keep on increasing the strength until the strength, it cannot act anymore. So it means that you just need to take your medication properly, so that now you keep those either bacteria out of your body, or you keep those viruses at a specific level in such a way that they are suppressed, so that you become normal. So when you keep them suppressing, now even time will come that even if you have sex with someone, it will not contaminate that other person. It means that you will be safe. Yes, you will have it, but it means that even if they test it, it will react negative, meaning that you are fine. Yes. But if you don't take the medication right, then it's going to happen that. So this topic is called present day evolution. The evolution which you can see. So this is what I was talking about. The HIV resistance or then lastly is human evolution. So after that, we're going to do some questions. We're going to mix them, and then we will conclude. Then here, these are characteristics humans share with other primates. Remember, I told you that here we mix. We can say that only this, or only on the head. Upright posture, large brain, eyes in front, eyes with cones, you are able to see color, free rotating arms, upper arms, you have the teeth, yes, sexual dimorphism. You can see this is the man, this is the female, yes. And what is it for? Yeah. So that reproduction can take place. And they're saying opposable thumbs, flat nails. Yes, flat nails, not close. I won't talk about that. Yeah, I won't talk about that. Ladies, I won't talk about it. They're saying reduced sense of smell. Guys, there is no need for you to panic. You panic for what? I told you. You panic for what? When your mother is cooking. How can you be hungry when your mother is cooking? All right. What are the differences between humans and other primates? So we said the first one is large brain. So we look at the brain is large. So we classify it as cranium. The skull, not the skull, the brain box is big. Here the brain box is what? Is small. So they have large cranium, which means that they have more brain cells, which means that if they have more brain cells, so it means that they are more intelligent. So that's what it means. So we're gonna look at it. So they can ask you that how is the brain related to intelligence? So it is related to the intelligence. So how is the cranium related to intelligence? The bigger the cranium, the more brain cells, the higher the intelligence. Yes. And then these are remember that we don't give you this, we just give you the diagram and then we ask you to to explain. So you need to know that the humans have flat face, this one have sloping face. You have brow ridges, there is reduced brow ridge here. You have the protruding jaws, you check them, they're protruding if you cut them. Yes, here the jaws are not protruding. So we call this situation prognanthus. This is prognanthus, this is not prognanthus. So large canines, small canines. Foramen magnum is in backward position. Check. Here it is a little bit forward, né? But here is a little bit what? Backward. This one helps us to connect the spine vertically so that it's able to walk on two limbs. You see the large canines, here the canines are small. The pellets is U-shape. Here is like rectangular pellets. You have diastema, spaces in between. Here there is no spaces. So you talk about all those differences. Those differences were summarized here. Yes, large cranium, flat face. So you have them in your books. You have them in your books. If you don't have, you can download you can get the book for distinction material, this book by clicking on join. And then after that, you'll see a link where you click on it, and then you get the book to use it so that and they're saying that whereas bipedalism working on two limbs, hind limbs, né? Or two legs. When you use the word limbs, you talk about hind limbs, né? When you use legs, legs are legs. They can't be in front. Okay, walking on two hind limbs or walking on two legs. What are some of the factors? It is foramen magnum. The position of foramen magnum, it is in forward position. Look at this. For the one which is quadrupedalism, it is backward. You see, it's a little bit back, while this one is a little bit forward, so that the spine can be connected vertically. Check. Here the spine is a little bit slanting because of the backward, while here the spine is straight. Then number two, we can talk about, so foramen magnum is in forward position to allow the spine to connect vertically. Pelvic girdle is short and wider. It's short and wider. Do you know that it's short and wider? That's why I want to talk about it. Do you know the reason why short ladies are wider? They can carry the upper weight. That's what it means. No, it's not my words. It's words from the book. Don't ask me who wrote the book. Short and wider to support the upper weight. To support the upper weight. To support the upper weight. Short and wider. Support the what? The upper weight. The spine is curved. It's curved. Meaning that it's shaped to act as a shock absorber for flexibility. Flexibility. So these factors, they can ask you the factors which make you to be bipedal, or the difference between the organisms which walk on two limbs and organisms which walk on four limbs. This one is four limbs, which one is two limbs. So you just look at the position of foramen magnum, S-shaped spine, and then the pelvic girdle. This is short and wider. It's short and wider, while this one is long and narrow. Yes. Why do we walk on two limbs? What are some of the advantages? You can hold the things freely, free arms, stand upright, you are able to see far, movement becomes easier, you remove the body from the exposure to sunlight, so all that. So these are some of the questions, but I have specific question I want you to attend it to. Lastly, but not the least, and there are some of the evidences which show that we have a common ancestor, and they are the same evidences which we should use when you are describing the out of Africa hypothesis. Yes, so we have the fossil evidence, genetic evidence, and then cultural evidence. So the fossil evidence, you talk about the fossils. Genetic evidence, you talk about mitochondrial DNA. Please don't use Y chromosome. It's no longer being set. And then you have cultural evidence, use of what? Use of tools. So we go to fossil evidence. Fossil evidence, wow, we can look at this. We call it, this is the transitional fossil. It has characteristics of this ancestor and the recent species. The ancestor and the decent what? Species, or the species which is present. So they can ask you what is the difference just by looking at two skulls. Yes. So you tell us that this species has both The tail is not as long as this species X and is not as short as this species Y. So when you are giving the differences, you have to explain, you link them together. Then these ones you have to memorize them. But it's better you need to know the following. Adipithecus means ad. Yes, that is the oldest. Australopithecus afarensis, we have Lucy. Yes. Then Australopithecus africanus, they can ask you, give two Australopithecus africanus species, you know. You have Taumchild, Littlefoot, and then Mrs. Place. Yes. Or they can ask you, give two Australopithecus africanus species, Australopithecus species, meaning that now they just look at Australopithecus. So you need to know the name, the scientific name, and the common name. The person who discovered them and where they discover them, in most cases, it come in objectives. So you need to know them. Yes. So this table is a little bit simple. You still one full day, guys, to catch up. Australopithecus sediba, this is called the carabo. Yes, it's carabo. Carabo is sometimes also called as a transitional fossil. Homo habilis, this is the handyman, the first individual to use tools. Homo erectus, erect. What does the word erect mean? Upright walking man. This is the first man to walk on two limbs. Yes. Homo sapiens, the wise man, the modern humans. It must be the modern humans. Yes. So the last is the same thing. The out of Africa hypothesis is the evidence to support the out of Africa hypothesis. It says that modern humans originated from Africa and migrated to the rest of the world. Modern, the word modern must be there. If you don't put it, zero. If they ask you to state the out of Africa hypothesis. Evidences, don't say mitochondrial DNA tools. No, evidences is genetic, fossil, and cultural. So if they ask you the examples of genetic evidence, then you talk about mitochondrial DNA. They ask you the evidence of the cultural evidence, then you talk about the use of what? Tools. Fossil evidence, we say that here, fossil evidence. Here, when you are describing it, the out of Africa hypothesis, you describe, look at this. All this old, you see it is Ethiopia. This old, you see this, all this is Africa. This old, all this is Africa. This old, up to here, all of them were discovered in Africa. Meaning that all the oldest. So that's why here you have to tell us that, you have to tell us here that, you have to tell us here that fossil of Adipithecus only found in Africa. This word only is a key point. Fossils of Australopithecus, you can give us examples, maybe Littlefoot, Taumchild, whatever, Carabo, you can give at least two. Where and the Homo habilis and the Homo habilis, this word, this, those are key points. Were only found in Africa, you see. Then when it comes to Homo erectus and Homo sapien, you say the oldest fossils. So the word oldest is a key point. So if it's possible, you can take a screenshot and then you are able to remember it. Genetic evidence, this one, we link it to that, the mitochondrial DNA, the oldest female was discovered in Africa. And that is called mitochondrial Eve. Guys, I'm getting late. Cultural evidence. Cultural evidence. Here we look at the oldest tools were used by the Homo habilis. The rest, we don't want them. The oldest tool were used by Homo habilis. We call it odd one tools. Yes. And then if you want to link them, that these tools, they were modified. As they are modifying them, even the brain also increased. How is the brain linked to the use of tools. These tools, to be made, they require someone with a bigger brain, meaning that the tools became more sophisticated as the cranium capacity or the brain size increases. So you see that as the tools are modifying from the simple to a more complex, you see that even the size of the brain also increases. That's what it means. All right. The last one is the phylogenetic tree. You need to know these questions. What is the name of this diagram? We say that is the phylogenetic tree. How many genera? Genera is the first name, meaning that you count only the first name. Let me come back here. You look at the first name. Adipithecus is 1, Australopithecus is 2. This one is repeated. Australopithecus, Paranthropus is 3, and then Homo is 4. Genera, remember to write the scientific name, you write the genus and the species name. So the genus, I've created a full video on this. Ne? Please, you can watch it. Make sure you answer all these questions which I put here for the phylogenetic tree. Make sure. What is the common ancestor of the species? So you look at which one is the common ancestor. Give two pairs of species which competed for resources, meaning that give two species which existed at the same time. You look at, you can just draw a line like this. Then you see that this one, this one, this one, this one, this one, they existed at the same time. Yes. And then you can say which species first walked on two limbs. Now it depends. In this case, this diagram, if you look at this diagram, it shows that the bipedalism started from here. So I draw a line. But if the question doesn't have this scale to show these things, then the first organism to use two limbs, it is Homo erectus. That's what we said. Then they're saying that which organism first work on two limbs. So we are saying that which organism first used the tools. You look at it. Where is the tools? The other one tools, they are here. So meaning that they started from here. You check. They started from here. The tools, the other one tools, they started from here. You see, maybe Paranthropus ethiopicus. Then if sorry, if they're asking you, they're asking you which organism. So we are done with that. Which organism? Where are we? Where are we? So if the scale is not there, then the organism which first used tools is Homo habilis. We know that. How many million years did Australopithecus africanus appeared? You look at where Australopithecus africanus. How many million years did Australopithecus africanus appeared? So you look at where did it appear from. It is here. So it is around 2.9 million years ago. Remember, we count from top going down. Yeah, remember this scale is million years ago. You see, we count from top to down. Or in most cases, we count from this side going that side. That's why this one is there. It has different scales. You see? Yes. You see now this one, we count from this side going to that side. The other question they're saying that when did the Homo habilis become extinct? How many? How many? They're saying that when did the Homo habilis became extinct? You look at Homo habilis is here. Its bar ends there. So you look at it here. They're saying that how many million years did Australopithecus africanus existed? So in this case, you look at what you want to calculate, meaning that, for example, Homo habilis, it started from there and then it ended there. So you get the difference between the 2 points. That's how long it existed. If you're talking about this and then this, that's when you get the difference between the two. Then they're saying that how does the brain size show evolution trend? Meaning that the bigger the brain, the more brain cell, the higher the intelligence. Meaning that now if it's about tools, they can be modified because these tools require what? Sophisticated sorry, they require more thinking. Then give the example of transitional fossil. We said that it is called transitional fossil Australopithecus sediba. Actually, even Homo naledi is a transitional fossil. How did the dentition show the diary? Meaning that if you have if you have this kind of teeth, it means that you are feeding on the rough tough food. If you have this kind of food, it means that you are feeding on the rough tough food. Check. Yes, you are feeding on the rough tough food. This kind, eh? Yes. So this one means that you are feeding on cooked food. So guys, I think let's just do some few questions. If you have more questions, let's look at page 9. The diagram below shows A and B shows the ventral and the bottom of the two skulls. Which diagram represents the skull of bipedalism? Look at the position of foramen magnum. It is in forward position. This one is backward. So the answer is B. Give visible reason for your answer, meaning that the position of foramen magnum is in forward position. You understand? Yes. In B. Tabulate the difference, visual difference between upper jaw of diagram A and B. Tabulate. Make sure that you draw a table. I've explained this. Make sure that you do that. Upper jaw. So they're saying the upper jaw, meaning that you only look at the jaws. You see? So meaning that this one is rectangular, this one is U-shape, and then this one has large canines, this has smaller canines, this one has even the diastema, meaning that the spaces between they are there. If you look at it, there are some spaces between. So you look at it. So in this case, they are talking about what you see there. So don't talk about flat face. There is no face here. Explain the significance of the shape of the spine that is associated with B, meaning that this, remember, it is S shape. We say this is bipedalism, you see. So it is S shape to allow flexibility of this organism. Yeah, to allow flexibility during locomotion. So that question is done. Let me look for you another question. Another question. Okay, I'll go to that one you're looking for. Okay. Okay, page 31. Thank you. Okay, now they are looking at this. They're saying that write down the diagram that represents the skull of bipedal. So if you look at it, look at the position of foramen magnum. You'll find out that here it is more of forward position. This one is a little bit back, and then this is a little bit moderate. So I will say that it is Z. You understand? Write down the letter of the yes. And also this they only say the skull. Okay, pelvic girdle of the write down but if you look at this, as I say, that this one is more forward position, this one is too much back. This could also be walking on two limbs because it's a little bit forward. So I think that's why they put two marks here. So it's gonna be Z and X, but Z is the most obvious. Yes. Then they are saying that Olivia, do you find your when you join, you scroll down. You see, just scroll down. Check there is there at the beginning, where you see that get the books. Yes. Then they're saying that describe how the structure difference between bipedal. Okay, give a reason for your answer. Give a reason for your answer to question 3.51. Why do you say 3.15b? Verificado of quadrupedal. Oh, they want to, is this one. Why? Because it is long and narrow. Then lastly, give they're saying describe one structure difference between bipedal and quadrupedalism. Structure difference between bipedal and quadrupedal organisms. Remember, we said that bipedal organism, the pelvis is short and wider, while the other one is long and narrow. Bipedal organisms, the spine is S shape, the other one is C shape. And then the bipedal organisms, we say that the position of foramen magnum is in forward position, then the other one is in backward position. Guys, position of foramen magnum is forward position. We don't want any other story. Then another question. Another question is the last page. Last page. Now check this question. They're saying that which species, they're saying that, you see now this is a phylogenetic tree. Which species in the diagram above existed or survived for the longest period of time? You have to look at it. Which bar is longer? You see that is Homo neanderthalensis. Is it Homo neanderthalensis? No. It's Homo erectus. It's Homo erectus. Because this one is for a Homo erectus. You see it started from here and then ended here. Yes, not drawing it straight, but you see where I'm talking about. You see what I'm talking about. Then the second question, they are saying that calculate the period of in million years of Australopithecus afarensis and the Australopithecus coexisted. Show your working. Australopithecus afarensis. Australopithecus afarensis. Yes. And what? Australopithecus afarensis and what? Calculate the period in million years in which Australopithecus afarensis and Australopithecus africanus coexisted. What time? How long did it exist? Afarensis is here. Afarensis is here. And Africanus is here. Meaning that this is afarensis and this is Africanus. So it means that you see that Africanus started here and afarensis ended there. Meaning that this is the period they existed together, coexisted. So the question, meaning that I'm gonna check here, you need to find out the scale and then get it. Maybe it is 3.4 and then 3. This is 3, for example. I'm just you have to calculate here. So that now you say 3.4 minus 3, so that is going to be 0.4 million years. So you see, name the species that was also known as handyman, that is Homo habilis. Name two uses of tools that was developed 2.6 million years ago. Where is it? Remember, this one was for, they gave you the answers. This one was for, they gave you the answers. This one was for, they gave you the answers. So now they are saying 2.6. Did I say 2.6? Developed where? Name two. Not that one. Name 2.6 million years ago. 2.6. So this is 2. So meaning that I'm going this side. The tool which was developed, meaning that this is the tool they are talking about around here. Remember, we start from 0. 0 is there. I'm going that side. So 2.6 is that side. So they're talking about this. They are saying that identify two species that use the most complex tools, meaning that two species which use the most complex tools. These ones, They use the most complex tools, that is Homo neanderthalensis and Homo sapiens. And then they're saying that explain how the change in the brain size over time led to the development of the tools. So increase in the brain size increased the development of the tools, because these tools, they require thought to develop them. Yes. Then the next question is about state. What is state? State the genus of A. Sediba. It is Australopithecus. So they want you to tell us that A is Australopithecus. Yes, because the first name is the genus, the second name is the species. Describe the shape of the spine of Homo sapien. We say that it is S-shaped. It is S-shaped. Sediba is thought to be a transitional fossil. State the meaning of transitional fossil. Is a fossil which has both characteristics of the ancestor and the most recent and the decent species, meaning the species which is still existing. Give two, we saw it with that diagram. Give the letter of the pelvis that would be representative of the Sediba. So it means that Sediba is a transitional fossil. They said 2 letters. Did they say 2 letters? Give the letter of the pelvis, meaning that he's supposed to have both characteristics. This is long and narrow, too much, and then this is short and wider. So this one is not long and narrow and not that short and wider. So it is A. So it means that it's in between this and this. That's why it's called a transition. It is in between. Explain the significance of change in prognanthus. Prognanthus means the jaws, protruding jaws. Explain the significance of change in prognathism from Australopithecus sediba to Homo sapiens. So here you explain why, because of the kind of the food they were feeding on. So remember that we said that the other one we are feeding on rafter food, raw food, and then this one they are feeding on cooked food, and then number 2 explain the significance. Yes, so meaning that the Australopithecus sediba, they didn't have that knowledge of cooking the food. You understand? Because cooking, it also requires you to know that, oh, there is fire, there is what, that you can cook. You understand? So that's how you supposed to explain it. Yeah. Then the last question, let me look for you a question here. Let me do. Wait, guys, I'm not going to do this question because I'm late. 38. There's that question. Take a screenshot, practice it. And then let me just do this one for you about Darwin. Looking for a question for Darwin. I prepared for you. Ish, man. Yes. Okay, there are 2 questions here. I prepared for you. Okay, I think you are done with the screenshot. Okay. Yes, the first question is here. Let's do it for you. Give it to you here. Mouse. Page 22. Good. They're saying that mutation occurred in a population of squirrels. Population was separated by a river, you see, that is speciation. Many years later, they discovered that the original population had undergone speciation. This process speciation is known as, diagram. What is a population? Population is the group of organisms of the same species that can interbreed to produce fertile offsprings. Yes. other than mutation, give other three causes of variation. You see, random mating, random fertilization, crossing over. Explain why there are eventually more squirrels with mutation on one side of the river. On one side of the river, where is mutation? Yeah, meaning that those ones were being favored. They are being favored. Those squirrels without imitation were not favored. Therefore, they died in large numbers. While those ones which had the mutation, they survived, and then they passed their genes to the next generation. Meaning that you're explaining using natural selection. You see how the question come? Explain the effect of the process above that on the biodiversity to the ecosystem in the ecosystem. Because some organisms are going to die and they can lead to extinction. Yes, they are going to die. They can lead to extinction. When you disturb the population, that's what happens. Then they're saying that it could be discovered that A and B were two separate species. Describe what can be done to confirm that squirrels belong to two different biological species. Describe what can be done that squirrel belong to two biological species. What can be done to show that these ones and these ones, they are totally two different species? You allow them to interbreed, and then they won't be able to interbreed to produce a fertile offspring. That's what it means. So you see that someone is asking me, am I not using comment section? I'm using. There is no one who is saying Sabero. I see you. I see you, Sabero. Saberu, why aren't you using comment section? Which comment section are you talking about, Saberu? Saberu. Okay, guys, the last one. They're saying that last one, the last question which I want you to talk about is here. So speciation, I've given you guys. Make sure that you do it. They can also bring it and then ask you to explain that process. So let me just wind up with this. Yes. That is our last question. They're saying that use the Darwin theory of natural selection, explain the evolution of lizards with the large jaws. You understand? Lizards with the large jaws. So there was a description whereby now here you're supposed to use large jaws so that you're able to explain. You see that now you're using Darwin's theory, meaning that what were the large jaws doing those small jaws couldn't do. Therefore, they survived, they passed their genes to the next generation. So now the question is not about Darwin's theory. Sorry, it's not about giraffe. It's about lizards with large jaws. So it means that you have to look at what made the lizards with the large jaws to survive. Yes. Then explain out of Africa hypothesis is an explanation of evolution of modern human. State what is out of Africa hypothesis. Modern humans originated from Africa and migrated to the rest of the world. That's what it means. Name the family to which modern humans belong. It belongs to family Hominidae. What genetic evidence is used to support out of Africa hypothesis? You see now they're looking for the genetic evidence, the evidence they have given you. So it's mitochondrial DNA. Yes. Describe how fossil evidence is used to support out of Africa hypothesis. I told you, I gave you the actual thing you're supposed to talk about. That fossil of Adipithecus only found in Africa. Fossil of Australopithecus, you give examples. Taomchad, Littlefoot, Sediba, and the fossils of Homo habilis were only found in Africa. Then you go to the oldest fossil of Homo sapien and the Homo erectus. The oldest, those two, you add the word oldest, were found in Africa. That's it. And then you get all those marks plus even excess. So guys, I think I'm going to stop here. I wish you the best. Thank you so much, guys, those who have been online asking. Sabelo, I see you. Thank you so much, guys. Thank you so much, guys. Erio, thank you so much. Jean, thank you so much. Rollins, thank you so much. Susan, thank you so much. And Lovus, thank you so much. Nomfundo, thank you so much. Winnie, thank you so much. Zintle, thank you so much, guys. Olivia, thank you so much. So, Akona, we see you. Sunflower, hey, there is also Prinisi, we see you guys, ne? Sabelo, we see you guys. Sherini, guys, make sure that you go and smash the paper. George, ne? Make sure that you go smash the paper. Don't forget that. When I said about the percentage increase, percentage decrease, don't forget the graph. Make sure that you do the techniques I gave you in paper 1 are the same techniques which is supposed to be used. Remember that the percentage is final minus initial over initial times 100. Yes, please don't forget about that. Don't forget that when it comes to draw a pie chart, you convert everything into 360. For example, you have 10, 20 and 30, maybe they are giving it in different style, so add all of them. After that, you say 10 out of the total times 360, times 360. If it is in percentage, you do the same. 10 out of 100 times 360, you get the degrees. Use the protractor, you draw a proper pie chart so that you are able to get the marks. Guys, I think that's it. If you have any question, you let me know. We love you so much. Thank you for subscribing. Thank you for sharing. Please don't let me down. Go smash the paper. I repeat, go smash the paper. The paper is supposed to be smashed. Yes, I think that is it. Stay blessed. See someone saying, oh, thank you, thank you, thank you. Until we meet again.